in some cases, we have to rely on the few plants available in collection or nurseries. So we have also some of these varieties are represented only by one replicate, so we cannot retrieve solid information. However, we have a large number under screening since 2015, as you can see here. What we observed after the first 2-3 years from the plantation and from the infection, we started to see some desiccation. However, at that time, it was not possible to categorize soon after 2-3 years the cultivars, because as you can see, we have some scattered desiccation. They were not uniform on all the replication of a given variety. So if a plant was not showing any symptoms, the question was, is that resistant, or is because still the incubation did not allow the onset of the symptoms. These plants were always tested annually. Sorry, sometimes maybe the picture are, okay, I'm sorry. And this is a couple of years later after the previous picture. You can see we start to see more prominent symptoms till now that we have a real good expression of the symptoms on the controls. And so we made the first attempts this year within the Bexil project and the national project to categorize all these cultivars that are under in the field. And in order to have some data that could be merged and joined with the genetic data, we classified the cultivars in these 6 classes from highly susceptible to highly tolerant. And the parameters that we used to classify the cultivars were the symptoms, so the severity of the symptoms, and then the bacterial load, how much bacterium was in the plants, because in general, when you have susceptible plants, you have lots of bacterium in the plants, as well as how many replicates for that cultivar got infected in the field. And okay. These are the results from one of the oldest field that we realized, the one that was funded by EFSA. As you can see, I put in green those that have a better response to the infection, and I mark the Lecino, which has the lowest percentage of infected tree, but also the lowest score of the symptoms, and the bacterial load is lower than compared to the other cultivars. For FS17, I marked I highlighted the fact that the plants, we had a very small number of trees. Even so, if you compare with another cultivar, susceptible cultivar, Don Carlo, which has the same number of replicates, you can see that a different behavior. And then we had the control Xylella di Nardò, which produced the expected results. So it's classified along with other cultivar as a susceptible cultivar. This is another plot, a second plot that was realized. Again, as you can see, the results on Lecino are always consistent. We have a low percentage of plant of Lecino infected with a high CQ value, so low bacterial titer in the plants, and also low expression of symptom. In addition, in this plot, we could identify another 2 varieties that had very low symptoms. I will show the picture later. Even so, you can see they did not perform as the Lecino because they had a higher percentage of infection and also the bacterial titer slightly higher. Then we collect the data on this cultivar, local cultivar Toscanina, which most likely we can classify as tolerant because it doesn't have symptoms, severe symptoms, but high bacterial load. And again, in this plot, the control was Oliarola salentina. Sorry, I put some animation, but they are not coming out. Okay, and below are in red are marked cultivars that behave like the susceptible Oliarola salentina, so very sensitive. Okay, these are some of the pictures that that comes from this that represent the data I showed in the graph. These are the symptoms that after 10 years from planting this Xylella di Nardò in the highly infected area, the symptoms developed. So we have a good susceptible control to categorize the remaining cultivars. This is on the opposite way the Lecchino, so without symptoms. And these are the Ascolana tenera, which is one of the cultivar that I mentioned that we recorded a resistant response in comparison with the Lecchino, and this is also Cipressino. Sorry for the slowness of the... Okay. And this is several cultivars performed as an intermediate. As you can see here, after 5 years, 10 years, Xylella di Nardò was almost dead compared to another local cultivar, Nociara, which is affected with desiccation, but in a less, to a less extent, and the same another cultivar, so you can see they are not resistant, but they are not susceptible as Oliarola. Also, in these two pictures, I want to highlight the importance of the incubation period. You see here Oliarola less affected than Xylella, but because the plant was 1 year younger. So as you can see, the incubation period for this experiment is very important. Then another aspect I want to remark is regarding the productivity of these plants. What we noticed in this plot for some cultivars like Coroneiki, Arbequina, and Arbosana is that even if they were affected by the desiccation, so they contain high bacterial titer and developed several symptoms, you can see they are full of production. So this is one of the difference compared to our local variety. Here I will go through other list of the cultivars that we have potentially identified as resistant, and as well as some that you see have score 1 or 2, which is highly susceptible. And in all cases, in all plots, Lecchino confirmed the same response and the same behavior. In one of the recent plantations, we tried to make this categorization just relying on the bacterial titer and the initial sign of symptoms, as for the incubation period was very limited. Even so, we could identify Lecchino as resistant and, for example, Coroneiki as a susceptible control. I have to say that if we cross some of the data between the different field and the greenhouse experiment, we have some conflicting data. For example, in the field, the cultivar Uovo di Piccione performed as a resistant cultivar, but when we inoculated the plant in the greenhouse, we had severe reaction as a susceptible cultivars, and the same is for this cultivar Giulia, which in our plot performed as resistant, and whereas if we have reports from commercial plots in which showed that the variety is not resistant. And the same was obtained, conflicting data were obtained for Coratina, which in one plot was severely affected, and in other plot, younger, was not affected. So maybe it's only a question, a matter of time. So the overall number of cultivars that are included in these plots, this is the separation based on three main classes, so the susceptible, the moderate susceptible or tolerant, and the resistant. As expected, we knew that the resistant traits in olive are quite limited, and you can see there are only 19% of this cultivar with putative character of resistant. One aspect I want to remark regarding the resistant cultivar is that when we go to see on the plants what happens in the distribution of the bacterium, we consistently find less bacteria in the leaves than in the stem in the resistant cultivars, and this is not the case of the susceptible cultivars. This is important because it has an important impact for the epidemiology, given that the insect feed on the young shoots or leaves. And additional data on the screening of the olive cultivar can be found in the poster of Professor Lima, and also you can enjoy the gift that was prepared with the olive oil harvested from these plots. And I want to say that collecting this phenotypic data is a real treasure for all the studies that are ongoing, because now we can merge this data with all the efforts that in the past years have been done to characterize the genetics of olive, and we know that there are several knowledge gap in the genomics of olive. And so for the first time, we can start to approach a GWAS approach to identify the mechanism and to narrow the genes that are involved in the resistance. And this is very important, because if we know the genes, we can speed up the process of breeding and screening of cultivars. I want to conclude in saying that this journey has not been only a scientific collaborative effort, but it has been very critical, the private support in this type of research. We need field, we need infrastructure that often we do not have within our scientific research center. So in this case, it was very important to have the availability of plots from farmers, and I want to thank the Azienda Foresta Forte and also the crowdfunding collected from the Merum and from Save the Olives, that is making a real strong effort to support our research, along with UNAPROL and more recently Filippo Berio, who are supporting our research program to provide support in the long term. That is very important. And I close with this. Thank you.
Thank you a lot, Maria, for keeping on time. Our next speaker is Silke Robaset from Ludwig Maximilian University of Munich. She will present genomic and molecular insights into olive immunity to Xylella fastidiosa, comparative analysis of Apulian cultivars, Ogliarola salentina, and Lecino.
Okay, thank you very much. I would need to change slides. Thank you. Okay, so it's my real pleasure to share some insights of our genome analysis in collaboration with Maria and also Pasquale Saldarelli on resolving the genomes of the Apulian cultivars Ogliarola and Lecino. And we analyze these genomes through the lens of the plant immune system, because this is what it's the core of the research in our lab, is understanding basically how the plant immune system works. And in principle, just to remind some people here in the audience, the plant immune system deploys two sets of different immune receptors. We have receptors that are called pattern recognition receptors, or PRRs, that localize to the cell surface, and these receptors mediate the recognition of common microbe-associated molecular patterns or damage-associated molecular patterns that are released from the plant cell during microbial invasion. And then this results in a broad-spectrum immune response. Now, successful pathogens would deliver so-called effectors, which are virulence proteins inside the plant cell, to suppress this level of immunity. And then plants deploy yet a second set of immune receptors that are in the cytosol, referred to as NLRs. And these NLRs then can detect these effectors or the actions of these effectors and then mount a much higher immune response. So typical common microbial associated molecular patterns would be from bacteria, the flagellum, and the derived immunogenic peptides FLAG22 and FLAG2-28, as well as the cold shock protein CSP22. And then a typical pattern from fungi would be chitin, and a typical self-damage pattern, which is also present in some microbes, can be the scoop peptides, or in microbial-derived scoop peptides. The well-known receptors, the PRs that detect these bacterial-derived or fungal-derived structures are FLS2 for flagellum or FLS3 for the other flagellum peptide. Then we have the core receptor for the cold shock protein, the SERC receptor for chitin, and then the MYC2 receptor for scoop peptides. So we resolved the genomes of the resistant Leccino and the susceptible Olarola salentina cultivar at chromosome level and at haplotype resolution, which means they actually have we have 4 genomes. And what's interesting to note is that the haplotype 1 from Leccino is most similar to the haplotype 1 from Olarola, and the haplotype 2 from Leccino is most similar to the haplotype 2 from Olarola, suggesting that they actually are derived from common ancestors. So through the lens of the immune system, as we hypothesize some of the observed resistance in Leccino might result from differences in the immune system, we then used computational tools to predict the numbers and types of PRRs and NLR receptors. And so you can see the different classifications, and overall, between the 4 haplotypes, in terms of numbers, there's not much of a significant difference. However, when we looked and zoomed into the LRR-RLK class 12, so these are leucine-rich repeat receptor-like kinases that form a specific subclass in the receptor kinase family, referred to as class 12, we noticed that both haplotypes of Leccino have significantly more of these receptors than the haplotypes of the Olarola genome. And these are encoding functional PRRs, and of course that could explain maybe, or is the hypothesis, that could explain some of the resistance that we observe in Leccino. So we zoomed into this class 12 family and developed some phylogenetic tree analysis here. And I hope you can appreciate that across all of the 4 haplotypes, we see a large expansion of 3 immune PRR clusters. So we have an expansion of the core-like clade, which is the receptors that have capacity to respond to cold shock protein. We see an expansion on the MYC2-like clade, which are the receptors with the capacity to respond to the scoop peptides. And then we have yet another clade, which is clustering with Peru, which is a receptor that detects an O-mycete peptide. What we also noticed is that the FLS2 receptor, which is a receptor most common across all land plant species and mediating the recognition of FLAG22, we only identify actually 1 candidate ortholog in one of the haplotypes of Leccino. So with that knowledge, we zoomed in and we profiled some immune responses in response to microbial patterns and bacterial extracts. And you can see here that in Leccino and Olarola, we barely see any response to FLAG22, which was surprising. So then we did some structure prediction of the 1 candidate ortholog from Leccino, and you can see that the olive ortholog is much shorter in its ectodomain. However, structure prediction of the immune signaling complex would suggest it is signaling active. But this is not being supported by MAMP-ML tool, which actually then predicts the immunogenicity of this factor, of this complex. So then that could explain why we don't really see an FLS2 response, FLAG22 response in Latino, or maybe only weak. So then we zoomed into chitin and we see very strong response in Olearola and a weaker response but significant in Latino. And again, family analysis supports this, that we have clear orthologs in the LIG4, LIG5 and SIRC1 receptor clades, which are receptors binding to chitin. When we clone these receptors, we see, and then express them in Nicotiana benthamiana, we see that the Olearola variant mediates a much higher chitin response compared with the Latino variant. So that might actually explain why we see a much stronger response to chitin in Olearola. But I say it's most important here for you guys is that we see a very strong response in Latino in response to bacterial extracts from Xylella fastidiosa pauca di Dono, which is much weaker in the Olearola sample. Of course, now the quest is open, which receptors mediates this response. So we also did some synteny analysis, and we noticed in a particular region of chromosome 16, there is an expansion in both haplotypes of the Latino genome, which is missing an Olearola. Apparently, this is a region where we see extensive contraction and expansion. And in Latino, we see a duplication of this region. Now, coming back to this phylogenetic tree here, marked in this yellow circles, you can see that some of the PRRs that are aligned to the MYC2 clade and also the Peru clade are specifically encoded in this chromosome 16 region. And some of these MYC2-like clades, if you look at the outer circles, you see actually are highly expressed in symptomatic trees. So as a summary, we have some evidence that there's maybe a weak or no response in olive to FLAG22. There's potentially a role for core receptors, although this has to be tested since core perception is only found in Solanaceae plants. Where's a clear response for chitin perception mediated by SIRC1. And again, there's a quest open, which is the function of this MYC2-like receptors that is so expanded, given that the scoop-like peptides are only present in Brassicaceae. And with this, I would like to close and give credits to the people who did the work. The work I've presented today is done by the people in the green circles, which is Kasia Rubak, Constance Tisserand, and Xing Dong. And there's also two people from my team here that are presenting short talks and posters, which I like to advertise. In yellow circle, Philippe Vera, he's presenting work on the microbiome therapy to Xylella, and Elif Olkun, she's in the red circle. She's presenting our works on small RNAs from Xylella. And I'm very thankful for many collaborations also with Italian colleagues here, and great funding also through the book. So thank you very much.
Thank you, Silke. Very, very nice speech. And the question later, so now we'd like to call on the podium Igor Weber from University of Brescia, and he will talk to us about regeneration of olive crop after unprecedented epidemic of Xylella fastidiosa pauca in Apulia, key factors in the evolving epidemiology.
Hello, I'm Igor. I'm from the University of Brescia, and I'll present to you this work that is a collaboration between Basilica Caramia, CNR, is funded by the Big Seal project. So to start, more than a decade after the introduction of Xylella fastidiosa, it is still a relevant issue in Apulia and in Italy. Resistant and tolerant cultivars, they represent an important and essential tool for the replanting strategies, but they are not immune to the infection. So when we speak about regeneration, we cannot only talk about replanting plants. It's more than that. We must also consider the integration the conditions in which they are established, the vector pressure, the inoculum pressure. So our objectives here was to investigate the incidence dynamics of the Xylella fastidiosa under natural conditions in newly planted olive plants of resistant cultivars, and also try to understand how the field level factors such as vector pressure and other agronomic factors could impact the incidence and the epidemiology of these resistant cultivars. So to start, we selected 16 olive cultivars in the infected area in Apulia, where we considered basically 3 factors. The first one was the cultivar, so we had Lecino and FS17 cultivars. These were plants that at the beginning of the experiments in 2023, they had around 3 to 4 years old. Then we also considered replanting density, so we have 2 different planting densities, low density and high density for Lecino, high density and super high density for FS17. And soil management, so how the management of the soil to target the vector may affect or not the incidence of the infection in these plants. We assessed also the vector population in these areas to understand their potential effect on the infection dynamics in these plants. So we sampled the juveniles and early adults on the ground cover, and also the adult populations through sweeping nets on the vegetation, the ground cover vegetation, and also the usage of chromatrophic traps placed on the canopy of the olive trees to understand the abundance around in the area of the olive trees. We also investigated the incidence of the infection at 2 different levels. We have at field level, where we sampled the percentage of from 30 to 50% of the plants in each grove. And we also, for each field, we selected 6 plants and evaluated through these 2 years of monitoring the dynamics of the infection inside the canopy. So to understand if there was a significant difference between the cultivars, or if there was an effect of the vector density on the establishment of the infection within the canopy. So the main results we have for the juvenile and early adult stage sampled on the ground cover. What we observed is that there was a significant effect of the management of the ground. So mainly in the internal part of the field. So in the internal side of the field, there was where the management was performed. In the external part, there was not a management on that area. So when we observed inside the field, we detected significantly less juveniles on the ground cover in the tillage compared with mulching. And in general, we had lower densities inside the field than outside the field. This for Philenus spumarius. When we look at Neophilenus competitors, we did not observe a significant effect of the management strategies in this case, although we have some potential effect based on the numerical differences. For the adults, when we look at the samplings on the ground cover through the sweeping nets, we also obtained a significant difference between the samplings on the tillage and the samplings on the mulching fields, especially for the beginning of the beginning and the mid-season. While for the monitoring in the yellow stick traps in the canopies, we did not observe significant effect or significant difference between mulching or tillage fields. We observed a more variable response in this case. Now looking for the incidence of the infection in the plants, here we have all the 16 fields. The bars, the field bars are the first year, the 2024, and the strip bars are from the second year. So for all fields, we observe that there is an increase in the incidence of infection, and the levels of infection of the fields, they varied from 2% up to 68%. But it is important to mention that there was absent or no significant symptoms in these plants, even for the fields with high level of infection. Here you can also see that we classified the fields according to the vector density. So we have 3 classes, the high, medium, and low vector densities. So we try to understand if there was a significant difference in infection level depending on these classes. So first, the soil management, when we look at the effect of soil management on the incidence, we did not observe a significant effect, meaning that the mulching and tillage did not matter much for the incidence of infection. But when we look at the vector density classes, so if the fields with low vector density, they tended to present more or less percentage of infection, we did observe that low fields classified as low density, they tended to present lower infection densities, while fields with high, medium or high densities, they tended to present higher infections. So what we observed is this non-linearity in the relationship between the level of infection and the vector abundance in the area, in this case for the adults. Now looking for the within canopy infection, what we observed and that was interesting is heterogeneous and dynamic progression of the infection inside the canopy. So for the same branch that was detected infected in the first sampling period was not always detected infected in the next 2 sampling periods. So we observed this heterogeneity in the detection of infection. We did observe some difference between FS17 and Lecino. In terms of FS17, tended to present a higher percentage of the canopy infected at the end of the monitoring period than Lecino. And we did not observe a significant effect, in this case, of the vector density in the percentage of the canopy getting infected for resistant plants. So to conclude, we have some main take-home messages. We observed that infection continued in both resistant cultivars. We observed that they become infected. The infection is still rising, so it means that although resistant cultivars are important for the regeneration and restoration of olive crops, can still become infected, and so we must manage not only not only replace with resistant cultivars, but also take account of other factors in other contexts, such as the vector density present in the field, other local contexts of the inoculum present in the area and the surroundings, to understand how to establish a long-term performance of these plants in these regeneration strategies. Vector pressure also mattered, as we observed. Medium and high density fields tended to present, were more associated with substantially higher percentage of infections in the resistant plants. Soil management presented some mixed effects. We observed that in the ground cover vegetation, there was some significant effect of the management, but when we look at the percentage of plants infected, they were not most associated with the soil management. And to conclude, regeneration requires integrated pest management. So as I said, resistant cultivars can support regeneration and the restoration of the olive culture. But alone they cannot, we need to provide other management strategies, phytosanitary measures to allow a long-term performance of these technologies. And this also leads us to another conclusion, that is the need to develop decision support tools that can help farmers, stakeholders to select the best management strategies in this restoration, for the restoration of olive crops in Apulia, in this case. So that's all. Thanks.
Thank you very much. So the next speaker is Annalisa Gianpetruzzi, who sent her apologies and will be substituted today by Pasquale Saldarelli, sorry, in his speech.
Good morning. Good morning, everybody. So, our treasure, 190 RNA-seq libraries that we have produced from the start of the epidemics, from early or chronic infection, either from the fields or from artificial inoculation. We tried to exploit these libraries, this data, to answer to some questions to our infection. The first question is, does olive sense Xylella and how early? So from one of these examples, at 48 hours post-inoculation, Leccino has sensed the presence of infections, and as you can see from the number of differentially expressed genes. In another trials, in Oliarola salentina, this sensing occurred at 10 days post-inoculation. To answer other questions we are trying to answer is that exploiting all this data, are there recurrent pathways or conserved transcriptional response among the different cultivars and varieties? This is an example, a trial of artificially inoculated plants of Leccino and Cellina, and a couple also of spontaneous seedlings resistant, harvested and analyzed in chronic infections. That means 12 months, minimum of 12 months after inoculation. We found that there are common elements between the two cultivars. 11 pathways, metabolic pathways, are highlighted, are enriched, and these are pathways that belong to the plant immune response, protein phosphorylation, protein kinase, defense response, while Leccino has also some exclusively enriched pathway, which are mainly related to the remodeling of the cell wall biogenesis, as you can see. When we move to analyze the top upregulated genes, we found again a plant immune response, which you can see here from the upregulation of pathogenesis-related proteins, several, and a couple of genes, which is a putative AG45-like domain, which was very intriguing for me, and the downy mildew resistance 6-like. You can see that there is also one of the resistance lyase kinase that Silke Robacek mentioned before, that are highlighted, are upregulated in Leccino in the first graph that you have on the left. Another pathway, another example of trial is Arbequina and 3 resistant cultivars that were analyzed in artificial condition. And again, we found these 2 genes highlighted, this AG45 domain and this downy mildew resistance-like. So they are recurring. The explanation, our likely explanation, is that the plants need to control their salicylic acid levels, because this DMR6 genes is a gene important to regulate the salicylic acid levels in the plants, to make them inactive and then protect the plants from an excess of salicylic acids. Salicylic acids induce a ROS production, so reactive oxygen species are very toxic for the cell, and also protect the plants by producing lignin and antimicrobial. With my surprise, it was also this EG45-like domain, which was reported in Arabidopsis thaliana as a plant natriuretic peptide. It has a similar role that DMR6. It antagonizes the salicylic acid level. So again, the plant feels the need to control salicylic acid levels. In Arabidopsis thaliana, this PMP, this AG45 protein interacts with a receptor-like protein. Again, a protein mentioned by Silke as important for the plant immune response, which is PNPR2. And this PNPR2 is distantly homologous to several receptor-like kinase that we found again upregulated in the plant, this GSO1, ATG, and so on. So it's a sort of circle that we is starting to close in a certain way. We have parallel evidence of the involvement of these genes also because we have proteomic analysis. There is a poster outside that tell us that these proteins, not the genes, but the proteins are present in infected plants. What likely, I say likely because we need further confirmation, makes lectino resistance. Lectino has a down-regulation of cell genes, so remodel the plant cell walls to create an opposition to the Xylella that advance. And also induce the lignin deposition as also showed by the upregulation of this lactase multicopper oxidase. Lignin deposition is reported from our group as increased in lectino. And there are two other genes, again, involved in systemic acquired resistance that control the level of salicylic acid. For simplicity, I will indicate this with SAMT and CABP2. They catalyze an opposite reaction. And as you can see, SAMT is very induced. SAMT inactivate the salicylic acid by adding a methyl group and producing this methyl salicylic acid, which is the form that moves in the plants to transfer the systemic acquired resistance to distant loci, and in such a way inactivating also the excess again of salicylic acid. So our likely working hypothesis is that upon infection, lectino converts salicylic acids to methyl salicylic acids locally, and then protecting locally the tissues from salicylic acid excess, but it produced lectino much more methyl salicylic acid that diffuse systemically and allow to protect the plants to transfer this defense response to the distant tissues. So there is a higher ability of lectino to induce a systemic acquired resistance. And that is all. I was in time. Happy for this. Thanks. I want to thank all the group to which I belong. I'm proud of this, and also particularly Annalisa Gianpetruzzi. She is not here, but she was l'artefice of the work. Thank you.
We will continue now with Cinzia Montemurro from the University of Bari. She will present a differential transcriptomic response of 3 olive cultivars to Xylella fastidiosa infection.
Thank you for the opportunity to present our results. So, as you know, of course, the Xylella fastidiosa outbreak in Apulia was identified for the first time from my colleagues of different institutions in Bari since 2013. And what is the situation now? We know that there are 4 cultivars that could be defined as resistance tolerant, that are Lecchino, Leccio del Corno, Lecciana, and FS17. We know that the most effective control strategy has proven to be replanting of susceptible olive trees with the resistance varieties, especially in the Salento area. But we don't know exactly which is the comprehension of the mechanism of the infection and why a cultivar could be defined as resistant and another as susceptible. So for this reason, we made a work that has the aim to comparison the gene expression across 3 olive cultivars. The resistant Leccino, of course, the susceptible Oliva rossa, that is an autochthonous cultivar from Apulia region, and the intermediate resistance cultivar named Donna Francesca. We identified this cultivar by a large microsatellite analysis among the germoplasm of Apulia regions, and we saw that this cultivar was grouped together with Leccino, so we supposed to be a candidate for the tolerance. In this way, we made an exploration of molecular mechanism, and also the final aim is the identification of candidate genes. So 3 plants of each cultivar were inoculated, artificially inoculated with Xylella fastidiosa bacterial suspension, and after 12 months, when the infection was systemic, we started the transcriptomic analysis. The RNA isolation was conducted by leaf and partial tissues along the plants. So this is the workflow of the analysis, started from the RNA isolation, the library preparation, sequencing, quality control, of course, of raw reads, the alignment, the DEGs, so the differential expresses genes identification, the gene ontology identification, and finally the Mapman analysis. And here you can find all the tools, the bioinformatic tools that were used. So, starting from the situation about the load of bacteria, in these graphs you can find on the left the PCR cycle after 3, 6, and 12 months the inoculation, and you can see that Oliva rossa, that is the blue graph, is lowest, so a lowest CQ means on the other graph and highest presence of bacteria. And you can find that, you can see that Donna Francesca has an intermediate behavior from Leccino and Oliva rossa. After the gene expression analysis, what was the results? So the results was that we have the activation of different transcriptional pathways in the 3 different cultivars. But the main results is that Oliva rossa, that is the susceptible ones, after the infection starts a wide reprogramming machine. In fact, you can see that there are more than 800 upregulated genes, and in the contrary, 600 downregulation genes. The situation is totally different in Donna Francesca and Leccino, in which after the infection, there are few pathways that are activated. So this is the first results in which the susceptible try to make in field all the instruments that have to contrast the infection without any results, while Leccino and Donna Francesca activate only some specific pathways. Here there is the functional annotation and the pathway, and in Leccino we have a few upregulated gene sets, especially related to ATP assimilation and sulfurylase activation, while in Oliva rossa there are a lot of pathways upregulated, many pathways related to infection, bacterial presence, stress, and so on. In Donna Francesca, generally there is a downregulation gene set of very few pathways, means that the response is similar to a constitutive response to the infection. In the Mapman analysis, we can find that Licino has a significant activation for the pathways related to metal handling, to hormones, and in general through stress, biotic and abiotic stress, while in Donna Francesca and Oliva Rossa, these pathways are down-regulated. So at the end, we defined a list of 9 candidate genes, choosing ones that are strongly expressed, overexpressed in Licino, and down-regulated in the Oliva Rossa. And for these 9 candidate genes that you can find in this slide, we set up a PCR, a real-time PCR, to validate the quality of RNA transcriptomic analysis, and we validated all the 9 genes with a strong expression in Licino, a weak expression in Oliva Rossa, and an intermediate expression in Donna Francesca. So at the end of this work, our results suggest that the comparative transcriptomic analysis revealed different patterns of gene regulation and defense strategies in the 3 cultivars. The Oliva Rossa, the susceptible one, exhibited extensive transcriptional reprogramming, but we saw, because it is strongly susceptible, that these responses were largely ineffective, while the moderately susceptible Donna Francesca retained a transcriptomic response with a weak activation of classical defense pathways, indicated that this intermediate level of susceptibility probably is likely mediated by constitutive defense traits. The resistant Licino demonstrated controlled and highly targeted transcriptomic reprogramming, so we can define it a precise defense responsive in this cultivar that minimizing collateral disruption of cellular process. This work is still continuous because these 9 genes now were totally sequenced, cloned, and now are ready to be posed in Nicotiana tabacum model plants for complementary analysis and to discover if the gene function of these genes in response to Xylella fastidiosa is validated. Of course, it's impossible to do in olive for all the limitations. So you can find all the results and much more in this publication on BMC Plant Biology, and I want to thank all the co-authors of this publication, especially Monica Miazzi and Valentina Fanelli, and Nunzio D'Agostino for the bioinformatic tools. Thank you for the attention.
So we would like to call Donato Boscia, who will give the speech on behalf of Pier Federico Lanotte, who cannot be here today.
Okay,
and the
you.
title is on the slide, sorry.
Good morning. Okay, so this is a brief presentation of the poster number 34. Poster number 34 that is co-authored by myself and some of my colleagues of my institute, together with few agronomists, few technicians that are involved in performing in a campaign of overgrafting of monumental olive trees, as Giovanni Melcarne, Cosimo Leone, Luciano Zaccaria, and Francesco Specchia. To preserve ancient olive trees from the severe impact of Xylella fastidiosa, the one that we have in Apulia, the subspecies Pauca st53, attempts in the past year have been made to substitute the canopy of susceptible trees by top grafting science of the resistant cultivar. Top grafts carried out on already sick trees that has been in several cases done 10 years ago or more or less or more, had a high percentage of failures. However, observation of some graft of lecino carried out casually before the epidemic and in good vegetative states have led to the hypothesis that grafting as a preventive action, so before the trees getting infected, could help to safeguard valuable monumental olive trees. This is why the Apulia region founds actually grafting intervention with science of resistant cultivars. So a survey was conducted between late 2025 and early of this year in 4 sites of the province of Brindisi, currently included in the demarcated infected, but on the forefront of the epidemics and under high pressure of inoculum. So you can see one example here, and graft were performed between 2021, one case, and all the other cases in 2023, so 3 years ago, basically, when most of the trees were still asymptomatic, most, not all, although some may have already been infected. The majority of the trees top grafted showed vigorous canopies and no sign of bacterial infection, whereas trees in neighboring groves exhibited severely decimated canopy, affecting 100% of the trees. Laboratory analysis revealed a percentage of grafted trees positive to Xylella ranging from 0 to 50%, while all the surrounding non-grafted trees were infected 100%. You can see detailed in the poster. Occasionally, some trees, approximately 13%, developed symptoms, always correlated to the infection of the cultivar Oliarola, thus suggesting infections was already established before grafting. Nevertheless, the percentage of symptomatic trees in top grafted groves that was significantly lower than in the neighboring groves, where it was 100%, with symptoms being markedly less severe. Taken together, these preliminary observations support the assumption that grafting with resistant cultivar performed before infection can help to preserve and protect ancient monumental olive trees. And just a few pictures to give you the idea. Or grafted on the right and non-grafted, you can see part on the left. This is another case, grafted, non-grafted. That's all.
Our next speaker is Angelina Velar from the IFAPA in Córdoba, Junta de Andalucía. She will speak about the World Olive Germplasm Bank of Córdoba.
Good morning, everybody. I would like to thank the organizers of the EFSA conference for giving me the possibility to talk about the World Olive Germplasm Bank of Córdoba as a source for research on Xylella control. Our collection is maintained at IFAPA, Regional Institute of Agriculture, the Andalusian Institute of Agriculture, where from more than 5 decades it has played an important role at conservation, management, identification, and evaluation of olive genetic resources. With more than 770 cultivars maintained in our collection, the World Olive Germplasm collection may be an important source of diversity against abiotic and biotic stresses. And in the case of Xylella, we have used the diversity to select 80 cultivars representative of both genetic and agronomic diversity. And these cultivars have been evaluated in different national and international project under both controlled and field screening trials. Some of the results were presented by Dr. Maria Saponari. And the meaning of this evaluation is to identify tolerant or resistant cultivars to Xylella fastidiosa. At the same time, our diversity can also be an important source to obtain new cultivars through breeding. And in this sense, progenies of Xylella fastidiosa tolerant cultivars like Lecino and Favolosa, well known as FS17, at both controlled and open pollination crosses has been maintained. The problem is that the seedlings of these progenies showed long juvenile period and only 28% of them flowered before 5 years. And among the adult genotypes, only 9 of them showed a good oil content measured in dry matter. And all these set of genotypes is going to be evaluated under both controlled and field conditions. We also obtained a progeny of Arbosana and Lecino with the aim to try to combine the good agronomic behavior and the resistance of Lecino, and this progeny is being evaluated in 2 different sites in Spain. Trying to find a good solution to have Xylella fastidiosa free plant material, the cultivars of our collection have been evaluated for their rooting ability under different thermotherapeutic treatment. You have there indicated the treatment, but only few of them showed very low rooting rates and at low temperatures and times, which is not suitable to kill the bacteria. Most of the cultivars failed to root under this thermotherapeutic treatment. Another contribution of our germplant collection in the case of Balearic Islands in collaboration with both IRFAP and EFSA was to identify to find the genetic identity of the prospective trees that were infected. And as you can see, more than 240 prospective trees were identified, and we found 85 different genotypes, among them 31 cultivated ones. And I would like to draw the attention of one case, the case of the cultivar Cirujal, a local one found in many trees in this study, but it's also a cultivar found under different names from Syria to France, and it is called Safrawi. But the most important result, in my opinion, is the identification of 9 unknown local cultivars, which represent an untapped diversity, and that's a good news for us in this conference, because the today conserved diversity is tomorrow resilience, and that's something very important to maintain. I would like to thank my colleagues of Italy, Balearic Islands, and of Cordova for all the collaboration, and you for the attention. Thank you.
So next speaker is Giorgia Sportelli, who will speak to us about genome editing in olive from hydrogenic callus induction to protoplast-based platform.
Thank you so much. Thank you so much for this great opportunity. Today I will, I don't think that my presentation, now yes. So today I will briefly talk about what me and my group are doing in Torino about genome editing in olive from embryogenic callus induction to a protoplast-based platform. I don't know if you know, but future and urgent calls in olive research are trying to address the uncertain biotechnology of this species, and of course to overcome 2 of the greatest bottlenecks that you will find working with this crop, which is genome editing, and specifically clean genome editing, DNA-free, and regeneration. In order to do that, we are working with different materials, which are protoplast, calli, and in vitro plants. For what concerns the protoplast-based platform, what we do is taking embryogenic callus of cultivar picual, which was obtained from our partnering project from Malaga, and through an isolation protocol, very genotype dependent, we obtain protoplast, which are naked cells that don't have any cell wall. So in that way, the editing system will enter the cell and ensure one single event of editing. In order to do that, we use the CRISPR-Cas9 system, which consists in the Cas9, the molecular scissor that will knock out, the selected gene, and RNA guide, which literally guide the molecular scissor on the gene to be selected. And in order to do that, like I was saying, we use PEG, which is polyethylene glycol, an hydrophilic polymer which transiently change the permeability of the membrane, the plasmatic membrane of the protoplast. And in that way, we combine the two items in one ribonucleoprotein complex that will be degraded naturally by the cell. So we tried this technology in our PQL protoplast, and it's feasible. We demonstrated that actually it can be done, and we did that combining the Cas9 with the GFP. So we assessed the entrance of the editing system through a fluorescent microscope. So we can say that it's feasible and it's a technology that we can actually use. The thing is that if we can't regenerate anything from this protoplast, this would be just theory. I mean, a valuable proof of concept, but just theory. So we want to assess if actually we can regenerate something from this protoplast, but we don't know if actually it's feasible because it has never been done. So we don't know if actually we will have eventually a whole plant, an edited plant, but we are very working with regeneration trials, so we will see. And in parallel, of course, we want to enrich our protoplast-based platform using different cultivars, such as, for example, we're working on Favolosa and Kalamata, which are respectively tolerant and susceptible to Xylella fastidiosa, because of course, like in our future perspective, we want to edit genes involved in the defense response against pathogens. So we want to work with different cultivars, but what we are doing is actually induce callus from adult explants, and we are using the PQLs from our partners in project as a reference model to actually compare the embryogenic features of the callus we have. And I mean, we are almost there. So if you want to know more about what we are doing with genome editing and induction of callus, please check our posters, number 19 and 25. And of course, I want to acknowledge all the people involved in this project, starting from my supervisor, Dr. Irene Perone, alongside with Dr. Giorgio Gambino, and of course, my colleague Mario Nicotera, who is attending with me this conference here. So please, if you have any question, ask then at the session of question. And again, these were my 5 minutes. Thank you all.
Our next speaker is Daniel Cucullo, and he will talk about plant cell wall dynamics in olive cultivars with contrasting responses to Xylella fastidiosa.
Good morning, everyone. I want to thank the organizer of this conference for the opportunity to present my work. Today I will speak about plant cell wall dynamics in olive cultivar contrasting responses to Xylella fastidiosa. One of the main barriers in plant-pathogen interaction is plant cell wall. Plant cell wall is not only a physical structure exposure to different biotic stresses, but is also a sensory platform. Many pathogens release cell wall degrading enzymes to colonize plant tissue, and in turn, plants send cell wall damage and trigger immunity, including a complex cell wall remodeling, such as addition of gallant side chains to rhamnogalacturon 1 and modification of pectin methyl esterification that will be central later in this presentation. We focus on 2 olive cultivars with different resistance to Xylella fastidiosa, the Arbequina susceptible and Favolosa the resistance. The aims was to analyze their cell wall composition and their possible cell wall remodeling during infection in order to identify the trait to be associated to resistance. Olive stems were collected from epidemic area of Cutrofiano, Apulia, Italy, and when screening for Xylella fastidiosa presence using qPCR. Cell wall material was extracted and the monosaccharide composition was analyzed using anionic exchange chromatography. At the basal level, both cultivars exhibit high-level xylose, galacturonic acid, and arabinose. During infection, the resistant cultivar Favolosa showed an increase of galacturonic acid and galactose, while the susceptible Arbequina show a decrease of xylose level. No significant differences were observed in cell wall cellulose content between the cultivar. The cell wall fractionation revealed that in the resistant cultivar, galacturonic acid and galactose increase in pectin-rich fraction during infection, while in the susceptible Arbequina xylose decrease in hemicellulose rich fraction during infection. These results are consistent with the previously cell wall remodeling observed in the total cell wall. Subsequently, we analyze cell wall material ester content and perform ELISA analysis. Interestingly, infection induced an increase in methyl ester content in the resistant cultivar Favolosa, and while no difference was observed in the susceptible. Consistently, ELISA analysis show an increased amogalturonal methyl esterification and rhamnogalaturonal 1-galatan epitome enrichment in the resistant cultivar during infection. These results are consistent with the role of pectin remodeling in plant immunity. To validate in tissue the pectin change observed, immunolabeling analysis was performed using GIM7 antibody. GIM7 highlight IL-methyl esterified homogalturonan. During infection, the relative signal of GIM7 was stronger in vascular tissue exhalent in the resistant cultivar, while no difference was observed in the susceptible. To conclude, our data suggests the olive resistant to Xylella appear to be associated with cultivar-specific cell wall remodeling strategy. High homogalturonal methyl esterification and rhamnogalaturonal side change remodeling emerge as resistance-associated wall traits. These changes may reinforce cell wall, limiting bacterial spread, and reducing disease development. I would like to thank Professor Vincenzo Leonetti, Sapienza team, our collaborator at CNR of Bari, our collaborator at the University of Malaga. My work was supported by Rich XI and XI World Project, and my participation in this conference was supported by All in Waste project. Thank you for your attention.
And then we have Rim Tuati now from ISPA CNR, and she speaks also for Domenico Di Cosmo, and she presents integrated metagenomic beta-barcoming and culturomics reveal beneficial bacteria in Xylella microbiome resistant cultivar Lecino.
Thank you very much. Good morning, everyone. This study was conducted in collaboration between the University of Bari and the CNR Institutes, ISPA and the PSCP, and today I will present how we integrated three complementary approaches to characterize the microbiome of Lecino. Previous studies on the resistant olive cultivar Lecino have already highlighted several bacterial groups that may be linked to its resilience. However, this works focused only on describing which bacteria are present, and we still lacked information about whether these microbes can actually do and how whether they can be cultivated and tested in the lab for the development of biocontrol development. Okay. To address these gaps, we integrated three complementary approaches: metagenomics, metabarcoding, and culturomics. We analyzed 270 olive trees from 9 orchards with different level infection pressure across Apulia. Starting from Xylella mood DNA, shotgun metagenomics allowed us to characterize both taxonomic composition and functional potential of the microbial community. We found a stable core microbiome across all seasons dominated by Microbacterium, Sphingomyces, and Bradyrhizobium. Functional annotation allowed us to reveal traits associated with microbial competition and antagonism, such as siderophore and secondary metabolite biosynthesis, quorum sensing, and biofilm formation. In addition, co-assembly enabled the reconstruction of metagenome assembly genomes and provided genetic resources for further genome mining analysis. In parallel, we performed beta-barcoding on XLM-SAP, targeting the bacterial community, inhabiting the fluid where Xylella colonizes and spreads. This analysis identified 16 bacterial genera consistently associated with the XLM-SAP across location, including taxa with reported plant beneficial and antagonistic traits. Finally, culturomics allowed us to recover the cultivable members of this microbiome. We isolated about 500 bacterial endophytic isolates belonging to 96 species and 38 genera from XLM mood, providing real cultivable strains that correspond to the functions predicted by sequence-based observation. Overall, the integration of these complementary approaches allowed us to characterize the Licino microbiome from community to isolate level. The convergence of taxonomic, functional, and cultivation-based evidence identified candidate microorganisms and traits potentially associated with Licino resilience. Most importantly, this study produced two key resources. First, a large collection of cultivable bacterial isolates, and second, a set of metagenome assembly genomes. Together, these resources give us what we need for future functional validation and for designing synthetic microbial communities for the biological control of Xylella fastidiosa. Thank you very much for your attention, and I would like to invite you to view our poster number 41 for more details about our work.
So this is a record, a marathon of speakers that tell you everything we know so far from research about Xylella olive resistance. So now we have, thanks to the speaker, the really excellent work, now we have time for the question and answer and discussion. So I would say the speakers stay here and they should get a microphone in order they can reply to the question. And we have a first question here from the audience.
Thank you. I have a question for speaker number 3. He showed those trials with FS17 and Licino 8 each cultivar, and he recorded the counts of the insects and also counts of bacteria infection. I want to ask if they measure productivity data, because as it was also shown and reported in the first presentation by Saponari, even susceptible cultivars or of medium susceptibility are still productive. And I think that when we score varieties for new plantings, for recommendations, we need to have the yield data as well. So just if he has those data.
Thank you for your question. These plants are still very young, so we have we started with 3 to 4 years old plants, so we still didn't have the chance to evaluate the productivity of these plants. So it is an ongoing trial. It will continue in the long term, and then we will have the opportunity to evaluate also the productivity, which is in fact one of the most important analysis that we must do in order to understand also the impacts of these infection levels that we are observing in these resistant plants. But yeah, we still don't have this information.
More questions?
Okay.
On the left.
First of all, thank you very much for all the presentations. They are very, very great. I have one question from number 8 speaker for genome editing in olive. Yes. So we are here to try to implement research finding in accent implementation, right? Accent, real accent. So in terms of your editing genes, how do you consider it under the GMO very strict control regulation in EU?
Thank you. So you have to repeat the last part of the question because was very low. So
sorry.
No, you're welcome. Thank you.
Because when I see you edit your genomes, I start thinking about the genomic modification organism regulation in EU. How do you place your research in that framework?
Sorry, how do you
think your work will be the future of it?
Yeah, now it's clear. Thank you so much.
Implement it.
Yeah. Thank you so much for repeating also, and thank you for the question. Okay, so I don't have all the answer because if it was so, I will be a fortune teller, not a scientist, and believe me, I will earn much more money. So. Speak of this, I don't know actually the future, but I do think that we have to really think about what we can do with genome editing in this crop, because a lot of other crops, it's possible to edit, but if we don't work and we are talking about from the beginning of this conference, if we don't collaborate with each other, that will be not feasible even in the future. Speaking about what genes we can edit, theoretically, everything. Practically, no. But of course, we are trying to focus on genes that can really be useful. For example, genes involved in the defense response, but also biotic stresses. And I do think that we will achieve that. Maybe not now, but maybe we will see in the next, Xylella Conference, maybe in 10 years. I don't know, maybe we will talk about something like this, concretely. And now I don't know, it's just theory, but we are working for actually have something to do in the real future. Thank you. Finger crossed.
So I have a question for Maria Saponari. Maria, I have a question for you. So you mentioned in your presentation that you test around 400 accession so far. Can you hear? So you mentioned you test 400 accession. And then we saw from Angelina Bellay's presentation that the Germplasm Bank collection in Cordoba has 755 variety and 1,300 exception. From the talk today, what is the way to achieve a speed system for olive-resistant testing? What is the future for testing more variety and more hybrids, more accessions?
We hope that this preliminary, this first group, this first group of data can serve to build up with the genetic data set that in the last 3-4 years have been advanced for olives to go toward the QTL markers, and so to avoid in the future to inoculate plants or plant these plants in the infected area to observe their response. But we hope that in the near future we can predict the behavior of the plants based on these new molecular markers.
This is a question made to all the speakers. is about the concept of tolerance or resistance, because based on the data provided, it seems that the varieties considered resistant harbor significant levels of Xylella. So my question is, of course, it's clear that the symptoms are less important, but what are the epidemiological consequences of that? I'm talking from the plant pathology point of view, not from the production or breeder point of view.
Maybe Pasquale, do you want to start?
An answer as a plant pathologist. This is an old story that every time we should re-say. So an immune plant is a plant that does not host, it does not allow any replication of a pathogen. That is immune, that people forgot. Then there is a resistant plant, which is lecino. Lecino has a low level of bacterial content, low level that we regularly verify in the field and in the field regularly and show limited desiccation. Then we have FS17 that has been defined as tolerant, and this is also written in the phytosanitary services document of the region Apulia, because it contains a highest level than lecino of bacteria, and also limit the desiccation. And then you have the susceptible, in which both level and desiccation led to the plants to die. So I propose, and I hope that you rely on this definition to stop to speak about this subject, because it's we call trito and ritrito. I don't know in English. this is all. Epidemiologically, of course, there is a risk. You asked for a risk. It's a plant that is infected, so it can be transmitted. But Maria and others show evidence that there is a less transmission from lecino of bacteria because of the low level of bacteria.
Yes, there will be a paper in few days out that has been accepted and coordinated by the group of Professor Domenico Bosco with our collaboration and the group of Basile Caramia, in which we definitely show that from the field the acquisition on this resistant plant is much lower, significantly lower than from susceptible plants. And this is in line with the literature on sharpshooters regarding the bacterial load in the plants necessary to have an efficient transmission. So in the resistant cultivar is close to the threshold or lower than what is needed for an insect to acquire efficiently the bacterium.
I think this is a very important point to show the transmission efficiency from these tolerant cultivars. More questions?
Sorry? I would like, I'm here, I'm just moving, because it's a question for Igor Weber, and it's linked also to the discussion about resistant and tolerant, because we noticed that insects, Philenus, is actually acquiring with a low efficiency from these tolerant / resistant varieties. And I was going to ask to Igor, because you tell that in your presentation that obviously the control of the vector is still important even in with olive groves with these tolerant resistant varieties. But I am asking you is if theoretically we could achieve immediately a landscape dominated by olive groves with these tolerant resistant varieties. I think that most of the new infection that you got came from olive groves with susceptible varieties that are surrounding your experimental groves in the landscape. And so do you think that in this theoretical scenario of olive groves of tolerant resistant varieties, the control of the vector could be lower, in your opinion?
Yeah, thank you for the question. But for the epidemiology to happen, we need to have the bacteria, the vector, and the host plant. In this case, if we have a scenario of basically resistant cultivars, no inoculum, so if you don't have other susceptible cultivars around that can sustain the inoculum, or other wild plants that has inoculum, it is one essential step of the part of the disease triangle that we eliminate. So theoretically, in a scenario like this, we would not require such effort for the manage of the vector. But unfortunately, we don't have this scenario. And in fact, I didn't mention this in the presentation, but most of the fields, we selected them in areas that were surrounded by century-old trees that were infected, or we have also other resistant new plant cultivation. So it was very varied, the landscape. So what we understand is that, in fact, these infections come from the surrounding, and the scenario that we observe depends obviously on what we have in terms of inoculum pressure and also of vector pressure from the surrounding. So managing our specific area does not also mean that we can sustain a low level of population, because it depends also on what the others around do to maintain low populations. This is why we think that when we observe the samplings from the yellow traps on the canopy, we observe that maybe what is maintaining the population low even in tillage fields is maintaining the population high even in tillage field is what is coming from the surroundings. So I think it's a little complex to understand.
There is a question here, Alessandra, in the center.
Here? I don't know if you can see me.
So thank you very much for this amazing conference. And I'd like to complete the question about gene editing and which gene could be interesting. And so based on the knowledge regarding the Pasquale showed, and also Daniele and Cynthia, it seems that the cell wall is a big player or important player in Latino resistance. And so my question is, do you think, guys, that are there some genes that if we silence these genes, we could increase the lignin and reinforce the cell wall and make the susceptible variety more resistant like Latino?
I strongly rely on plant cell wall remodeling. I have also a specific project on it, because also from the Pierce disease, it is known that for example, the methyl esterification of the lignin is a factor that make the plant cell wall less aggressive, less digested by the bacterial enzyme that degrades the plant cell wall. So I would say yes, if there is a gene that could increase in certain way plant remodeling, but you should be careful because it's the xylem system. I think it's a delicate system that you can modify. But Of course, yes, the position of lignin is described for Lecino, not from us, from other people, and we start to have this evidence also of the strong involvement of this.
Cynthia?
Yes, I'm very happy to discover today that 3 different works have the same conclusions. So means that we are on the right side to identify the real molecular mechanism that can explain the resistance of Xylella. For sure, cell wall play a unique role in the answer. In our studies, we identified the overexpressed genes. Usually with genome editing, it's more easy to knock out the function gene, but there is also the way to overexpress these genes. So for sure, we have to discover if the real gene function is validated in the model plants, and after we can think about the genome editing approach also with new technologies such as ribonucleotide proteins and so on to make all the plants with these characteristics.
Okay.
Hi. I have a question for Maria. I was just wondering if, because you showed us, plants that when in greenhouses were represented as maybe resistant, but eventually became more vulnerable to Xylella infection when moved to a real case scenario. So I was wondering if there's a correlation, there may be any correlation with the rhizosphere-associated microbiome variation. I mean, if there are sequencing of the bacteria that are in the rhizosphere are affecting maybe the susceptibility to plants.
Yes, in the greenhouse, I would say that is easier to control some external parameters for the test compared in the field where really, even in the same plot, we do not have the same soil, and so we have variation that even in the replicates are affected by the soil structure, for example. We have evidence that, for example, in one plot where there was more water flowed compared to another area, the replicates behave differently than in some random spot. But in the greenhouse, we try to use the same soil and to have the same standard condition to minimize this effect. But it can be also that it is some the disformity that we observe among the replicates can be due to different condition of the plants. So yeah. Generally, for the susceptible plants, you have more conserved reactions. For the resistant plants, it's more heterogeneous sometimes the results. Then there is the factor of the artificial inoculation by which we put an overload of bacterium compared to the vector, so the conditions are not exactly the natural one.
Okay.
Good morning, everybody. My name is Sarah Brunel. So I work for the International Plant Protection Convention based in FAO, particularly in implementation, and I'm getting inspiration for preparing concluding remarks of this event. So what I really want to do is to find a coherence in between all the presentation during this session, in between the different sessions, because everything we're doing here is for a concrete purpose, which is to help people in the field to find concrete solutions. So my question to you all, we've heard researches on the microbiome, on the cultivars, on the cells, on the genes. Did you guys work together, share your results? So that maybe we can lessons learned from all these researches and a way forward. How do we use all of your findings into concrete and operational guidelines for areas where Xylella is present and areas where Xylella is still absent? And maybe that's also a question to Giuseppe. What can we do together to ensure we promote such a mechanism for implementation? Thank you.
So, who want to start? Maria?
No, I can say that the fact that the European Commission for the first time, I think, was for Xylella, funded a project devoted only to Xylella has allowed us to put together a multidisciplinary work. So if you go to the past project, but also in Bexhill, you can see that the different aspects that you mentioned are tackled together. And so there is a quite a long experience of a multidisciplinary approach to the problem. And we hope, until now, since 2015 until August, we had continuous funding from the European Commission. We hope that this will be continued to finalize the results.
If I can add to this reply, so olive plant resistance is a very long-term activity. It's not something you do in 1 year. What we see today is huge results, but it's results of 3 European projects and several national projects. And there was a very small project from EFSA beginning, but very, very small one. So we really need to put effort and to think long-term on resistance. And Xylella is affecting olive in Europe, particularly in Italy and in Balearic Island. Is also affecting olive in South America. So somehow the target is that the olive germ plant collection of Cordoba and other olive germ collection are tested all, and that different countries can prepare, because somehow with plant pests we know they are moving, so it would be better to prepare before, to test variety before they arrive than doing afterwards. And now, for the first time on Friday, we will go to south of Apulia, to Lecce area, and we will see the plantation of resistant, tolerant variety in the field, which is now becoming a reality, but took really a lot of effort and time and funding, and more funding is needed for this topic, and we should prepare also for other variety, other environmental conditions.
Hello. To follow what Giuseppe was saying and what Maria has been explaining today, I think that we should continue with the evaluation of olive cultivars, but both routes, either the traditional one, the evaluation in the field, but also the screening through molecular markers that Maria and her group will develop, because we need diversity in the field. One of the lessons that we have learned now is that if we do not have diversity in our fields, we cannot diversify the risk. And that's important not only for the present, but also for the future, because we were talking about crosses, and let us remember what happened to the high-density orchards, where most of the new olive cultivars are either daughter or stepdaughters of Arbequina. We do not want to have that for Lecino as well in the future, to say that most of the new olive cultivars that we have are either daughters or stepdaughters of Lecino. And at the same time, we have to combine the diversity with the good agronomic performance. And as it has been said here, we're talking about olive, and the studies are not easy to perform. But we can ensure all of you, because everybody knows that we work in a multidisciplinary team, and we have been trying. I am the last one to talk about Xylella, but I have learned a lot, and I hope that we'll continue to work together in the future. But it is something that it is a key for all of us, and we work on that in the present and in the future as well. But we also need funding, and FAO can also give funding. Thank you.
More questions?
I have a question. Maybe for Pasquale. So if I understood what you're proposing is like this a reduction of methyl salicylic acid as a response in the resistant varieties. Yeah. So how, do you see like a spatial response when you inoculate? The response is volatile, and then have you tried to inhibit these enzymes that move methyl salicylic acid to salicylic acid and vice versa to prove your hypothesis?
No, as I underlined, it's a likely working hypothesis now. For now, we have evidences that both genes that I mentioned, the four genes, the DMR6, the AG45, what I call AG45, the receptor-like kinase, and also the two that regulate finally the exchange salicylic acids and methyl salicylic acid. They exist and they are induced. Even some of them in parallel gene expression analysis. I have no evidence of the level of salicylic acid of these plants. I'm looking. It's an invitation for people here to help us because the dosage of salicylic acid is made by HPLC analysis, and we need a chemist that help us to check the levels of this. In opposition to what I say, to this working hypothesis, there are some old, made from Maria and other Enza Don Giovanni testing, field testing with acylbenzoar. Acylbenzoar is an analog. It should, in theory, stimulate salicylic acid's route. They were not successful in the fields, but in the fields we are learning now. You will see later in the session 2 that working in the fields is another world, another story. So, but this is a working hypothesis. I will try to follow, to pursue.
At the end, there is a question. Yeah.
Hi, thank you for all the research. I was wondering in terms of, so now that you mentioned we need diversity in the olives in the field, and also we talked about grafting. I was wondering also screening of different olive cultivars. I was wondering if you look into the wild populations of olives and if they can be used for grafting, or are they resistant or susceptible to Xylella fastidiosa? So what are your insights on the wild olives in that term?
Maybe Donato, you can answer. She's talking about the use of wild germplasm in the wild olives, if you can find resistance, if I'm not wrong. Yeah. Yes. In the wild, there is a source of resistance or can be used for regenerating the canopy.
Yes, one of the first approach before we started the crosses, because we knew that it would take at least 8-10 years to generate a new productive plants. What we did is I would say the farmers in the area helped us, is to look for healthy-looking spontaneous plants, and we found several that were potentially resistant. And what was interesting when we did the parentage analysis of these seedlings is that the majority of them derived from Lecino, so confirming the resistance. I have to say that it's not easy to find seedlings that are both productive and then suitable for an agronomic use. I have also to say that the infections in the wild genotype is quite different from the cultivated plants, so the plants are very vigorous, have a different type of leaves, so maybe less susceptible to vectors, so it's not only the genetics of the plants, but also the environment that was important.
We made a similar analysis on some plants that are present in infected areas that present no symptoms and a very, very, very low bacterial load. And in almost many cases, they are seedlings of Lecino and sometimes also Silvestris, so the wild Olea europea. We have also the evidence that there are some interesting plants in Tunisia derived from crosses from wild olives. There is also an interesting theory about the involvement of Olea cuspidata in the resistance of Xylella fastidiosa.
In Spain, in Balearic Islands, as I explained before, many wild genotypes were found, but we have to separate two things, as Cinzia was saying. We have the real wild, the real laster trees, but we also may have seedlings from spontaneous seedlings from cultivars or crosses between cultivars and the wilds, what we call feral forms. So if we're talking for real oleasters, as Maria explained, it's difficult to evaluate the resistance, but I'm not sure, but as far as I remember, the data that Diego was giving to us, you found diversity. You had resistant genotypes among the wild, but also susceptible ones. Our experience in verticillum was that they are not comparable, but the percentage of, if we talk about percentage, of resistant genotypes among the wilds was very similar to the one of the cultivated ones. And we have to bear in mind that the difference between oleasters and cultivated olives is not as in other crops. I mean, genetically they are very similar, and the genetic distance is not as big, or even the morphological differences sometimes are not so easy to realize. You can rely on the very small size of the fruit and on the fact that many of the seeds are really smooth because they have been transported by the birds. And that's all I can say about that.
Okay. One question, Elversio, here. In
the center.
Okay. I have a question and a comment. First, the comments. First of all, thank you for this wonderful event. It's beautiful. So my comment is about the tolerance, resistance, so on. Okay. Of course, that immunity, that it's a dream, but you don't have this immunity. Okay. So they use the Lecino to propagate the material. You have production, you have some symptoms, small symptoms, but you have the productions. The growers need production. Okay. Because, just for confirm the Lecino, I have the same results in Brazil, that Lecino is resistant to Xylella also, the same results that you have there. My questions now is about why the guys talk about the Lecino and they didn't talk about the FST, because just the comments comes from Lecino, not FST, like using those propagation in the field, thinking about the concept of the diversity.
Did you get the question? Can you repeat the question?
You have to shout a bit.
Okay, so my question is how the comments is doing about Lecino, okay? But AFST by the data is resistance also, okay? Do you understand me?
No.
No? So, the data that shows that the Lecino is resistance also. AFS17 is resistance also, okay?
It's more tolerant than resistant, according to the definition that we have.
It's dependent on definitions, point of view. Okay, but it's the same, okay, the same response that you have, okay? My question is, thinking about diversity, not using, not recommend AFS17 also, okay? Because I understand that AFS17 is also tolerant or resistant, depending on point of view. Okay, is that my questions?
Yeah.
So I don't know if he refers to the transcriptome, to the plant response of FS17. Is that right? Is that you are asking? If you ask this, we have a similar response regarding these genes that I mentioned before, but it's completely different. We have not a lot of experiences with FS17 in terms of plant response. I would mainly rely on Lecino, which was more deeply studied about this. Regarding these genes that I mentioned, the behavior is the same.
Yeah, I think also, Elvesio, that your comment was that they were recommending only FS17 in the region or more. Both, yes, but you have to have in mind that since a few years ago, there were the only two cultivars that proved resistance and consistency during the years. Now that the experiments are ongoing, I think with the trials that Maria and all the people are developing with the higher number of cultivars, new lines has been shown promising a similar level of tolerance or resistance than these two cultivars. And as we explained, there is still a long collection of germplasm ready to be tested. And we need to have in mind that it took more than 10 years for the collection, the first collection, to show symptoms. So this is still a very long process. We need to keep in mind that with the research, with the state-of-the-art techniques that we are using, we will speed probably in the next year the process of selecting new material, maybe with the gene editing and the new technologies, CRISPR, and the change in the mentality of the European Commission, how to approach these techniques. We will be able in the future to speed up the process of getting more material for providing the farmers that is productive. We cannot forget that the trees have to produce olives to make the people to survive and to get some economic incomes. And with this, I close the session. I really thank all the speakers. They keep perfectly on time. And thank you for all the nice questions, very interesting questions that you made.
So we are going for lunch. Lunch is 12:30 to 2 o'clock. And we will start at 2 o'clock sharp here with the next session on advance on control of Xylella. And during the lunchtime, for those who didn't do yet, there is time to hang the poster for this evening poster session. So if you have a poster not yet hanged, please ask at the reception how to do it, where to do it. And have a good lunch.
Sì, sì, la stessa, sì. He's in the pitch talk. Ok, that's the second page.
It's the second page.
Sì, bravo.
Okay. You want me to put some other...
No, it's okay. So I will wait for the general session and present Stefan. More than the room. I take the jacket. Just to sit there to invite all the participants. We wait for the roll to start. So we invite we wait for, faccio già un avviso? Ok. Salve. Good morning, everybody. Can you take the seat so that we can start with the presentation? And also I invite all the speakers to have a seat on the right of the... Have a seat on the right of the, on my right, also the people from the pitch talk. Prendila da là, prima c'era. Sono partiti. Madonna mia, 20 minuti. Okay, good morning everybody. Me and Anna Bonaterra will chair this session. I'm particularly happy to chair this session because it comes from a research field that is not so much investigated, the control. It's an hard field because it requires a lot of field work and a lot of years to get consistent results. So let's start and to have the first speaker, Stefan Compant, that will speak about the mode of action and field trials achievements of biopesticides for the control of Xylella fastidiosa or its insect vectors.
Thank you, Pasquale. Good afternoon, everyone.
We need the slides.
Yeah, thank you. So I think you know also this talk because you will participate in this talk.
Yeah, later.
We split half of the presentation together, and we would like to speak about some results about BioVexo, about the mode of action of some innovative biopesticides and the result from the field. So what was BioVexo? It was a European project granted by BBIGU, now CBIGU, and Horizon 2020. It started in 2020 and ended in December previous year. We were 11 partners, some from academia with RTOs, university, Also some farmer associations, some industry, and SMEs from different countries, Austria, Spain, Italy, Belgium, and Slovenia. Only 11 partners to develop some biopesticide to target either the Xylella or the insect vector, and our aim was to produce a new biopesticide. Why? To reduce the Xylella pressure, disease severity, to protect and safeguard olive orchard and secure job for the future, and a sustainable approach also to protect the landscape. How we did this project, we split in different work packages. There was one work package when we developed biopesticide targeting Xylella directly on plants, what I will call X biopesticide, X for Xylella. A second one to target the vector, Philonneus pomarius, V biopesticide, V for the vector, and there was a work package to upscale and formulate biopesticide before to go to the field and apply the different solution, putative solution in large-scale field trial and also in IPM strategy. I was scientific coordinator of this project, but also I was work package leader dealing with the mode of action. It means that if there is something working in the field, our goal was also to determine how it's work, what is part of the mechanism of action. Of course, we wanted to develop also sustainable biopesticides, so we made the toxicity and toxicology tested, and also life cycle assessment for each product, also the tests of the solution on non-target species, like for example bees. We didn't start from scratches. We had some different biopesticides at the beginning, starting at TRL45. Some were bacterial strain. One was Pseudomonas, PV8, Bacillus GG22, and Paraburkholderia phytophilomons strain PAGN, for which Stephen Lindo from California shown that it was effective to control Pierce disease caused by Xylella on grapevine. There was also microbial metabolite. We did not continue with this microbial metabolite. 2 plant extracts, one based on onion extract, targeting Xylella, and another one based on pelargonic acid, non-arenoic acid, from Globachem, targeting the insect vector. And there was also an anthropogenic fungus. Our aim was to try to develop solution for preventive approach, also curative, directly on the plants, on young and old trees, but also to fight the insect vector and to develop sustainable organic solution. The result we obtained so far after 5 years and half year of a project, there was a lot of tests done in the field. Pasquale will speak about that, but there was also upscale and formulation of different biopesticides. We tested also a spray or drip irrigation or endotherapy in the field. We performed the toxicology and toxicity tested, so life cycle assessment, and the mode of action. So at the beginning, there was 6 biopesticides, 3 targeting the vector and 3 targeting Xylella directly on plants, but at the end, we finished with 3 promising biopesticides and top-performing biopesticides. So at the end, we had onion extract targeting directly Xylella on plants. Sancari, now it's on the market. It's based on pelargonic acid targeting the insect vector, and also Paraburkholderia phytophilomons strain PAGN, developed by SS Bio in collaboration with AIT. This was a pack before to do field experiment. And I told you I was responsible also to analyze the mode of action of the different biopesticides. First, if the biopesticide can directly be antagonism against Xylella. Also, if the colonized Philonneus pomarius or colonized plants as an underphyte or epiphyte. Also, if they can boost the plant immunity. And so we study also colonization process, the persistence of a strain in the environment after application as foliar spray. Here you have plant colonization by Parabiocolderia phytopheromones to PSGN. I use a fish to show these microbes here as orange, what you see on the second panel on the surfaces of leaves that are blue of olive. It's confocal microscopy, and we detect it on the surface, never as an on-the-fight after spray application, also on the nerve. But what was interesting is that after 8 applications, there was a very low number of cells on the surfaces of plants when we made the test under greenhouse condition, but also in the field. We analyzed after application of biopesticide if the biopesticide could also impact the plant microbiome, so indirectly, and what we saw that there was a very small effect of strain PAGN, also onion extract on the fungal diversity, but also on the bacterial diversity. It looked like there is not really there is only a small impact, and what changed the plant microbiome inside plants is more due to the cultivar or the terroir where the plants were grown. We analyze also the mode of action, especially the team of Pasquale, what are the plant response to the different biopesticide. Onion extract, Sankari, it has been submitted, this publication, and what we saw, for example, is that some biopesticides, they boost the plant immunity, but also upregulate a gene related to plant growth stimulation. And now, even if I talk to you about mode of action of biopesticides, this was done in the lab for molecular biology, but we need also a result from the field, and I would like to pass the talk to Pasquale. It's yours now.
So thank you. I duplicate myself for the, as you learned from the presentation of Stefan, I was responsible of the work package 4, which deals with the field trials. So testing the biopesticides in field trials. And we did 2 categories of field trials, curative field trials. They lasted 4 years in 2 different places in San Vito, in the outbreak area, in San Vito dei Normanni and Oria. And you can see that in San Vito dei Normanni, we planted directly, and so we knew the history of these plants from the start of their plantation to the end of the trial. I would say the BioVexo is the only one trials which used such a long number, a big number of plants, because we worked on 168 plants of Xylella di Nardò randomized, and we monitored throughout the time Xylella by quantitative PCR and the symptoms every 6 months, going in the field, picking the tissues and observing the evolution, the progress of infection, which was a natural infection, natural condition. While the other trials was on old plants already showing symptoms. It was in Oria, and it was carried out on 120 plants. I immediately say that the products that we used, that means PSJN and also the onion extracts that Stefan mentioned before, the 2 biopesticides were not able to eliminate, to eradicate Xylella from infected plants. So plants get infected. There was a trend throughout the time, as you can see from the 2 graphs of the CQ value. There was a trend of formulated PSJN, particularly to reduce the CQ value, but we were not able to eliminate the bacterium from the plants. While we were able to mitigate the disease. This was, I would say, a good result, a starting results from this project. As you can see from the AUDPC progress, that means the progress throughout the time of the symptoms observed in the fields on a scale of 0 to 5, the symptoms were mitigated up to a 27% 27.7% by PSJN, the Parabullcorderia. It was not the same on the older plants in Oria, but also in this case we observed a mitigation of the symptoms. To have a further confirmation of the well-being, the good status, physiological status of these plants, we implemented such physiological studies on the same plants in the field in different cases by looking at stomatal conductance and canopy temperature. These measures were based on previous experience that we had and we published it, and we had also in greenhouse growing plants, telling us that Xylella infected plants suffer a draw stress induced by Xylella because of the closure of the stomata induced by the biofilm formation of the bacterium. So we observed that plants treated with formulated PSJN had a better condition, even significant of stomatal conductance. So treated olives suffer less the draw stress of Xylella. This was particularly evident in the younger plants, not exactly evident in symptoms in Oria. We conducted also field trials in Mallorca, again the same for preventive trials and curative trials, and we get lower symptom severity of PSJN and onion also compared to the untreated, but no difference again in the rate of infected plants. Then we implemented in 2 sites an integrated pest management. That means we combined the biopesticides to Xylella, that means PSJN and onion, to biopesticides against the vector, the pelargonic acid, which was mentioned by Stefan. The name of this pelargonic acid is Sankari. It's a traded mark. It's already in the market. We worked on 432 olives of the highly susceptible cultivar of Olearola salentina, and we did application against the juveniles, so an integrated pest management approach. And we did the control of juveniles form of Philenus spumarius and also the adults, performing the application when the flight of the Philenus spumarius was present in the fields. We used as a control a chemical control of the adults of Philenus spumarius by acetamiprid, as you can see in this graph. You can observe that in this first site in Francavilla Fontana, we get a reduction and a better efficacy in the percentage of the infected plants with an Abbott index, an index of efficacy of the combination of PSJN with the treatments up to 72% if we consider the plants treated with the chemical insecticide or Sankari, the biopesticides. With the biopesticides, you can see this in the frame where it is the Sankari adults written. We got a good efficacy also of containment, of mitigation of symptoms. The other sites, we did this, we replicate this in 2 different environments. In Squinzano, this is another place, and we again observed a reduction, a better control of the infected plants with the formulated PSJN. So our final outcome. We formulated PSJN was able to slow down the spread and symptoms up to the 27% of the efficacy after 4 years in curative plants, young olive plants, and improves plant vigor. It's a biostimulant. We observed these effects better on olives, but also on almonds. Onion extracts improves stomatal conductance and reduces the symptoms, new symptoms on olive and almonds. And Sankari, because I did not mention, but we did also studies to the vector directly in semi-field condition, is able to reduce the juvenile and insect adults of up to the 54%. In integrated pest management, Sankari was able, in combination with formulated to PSJN, to reduce up to the 40% the percentage of infected plants, while with onion we don't have efficient, consistent results. I need to thank also my colleagues from the Istituto Basilica Ramia di Locorotondo, in particularly Enza, Don Giovanni and his team, because they were responsible of the field trials, directly responsible in the field trials. I want to conclude telling that BioVexor represents the first, to me or to us, is a starting point. Perhaps someone after us will be able to do better than us, but it is a first starting point that was particularly hard from our side to carry out in the field for a period of 4 years. These data have been accepted for publishing in Plant Disease, a journal of the APS, American Phytological Society. If you want to know more, you can look there. And also the paper, the data of the transcriptome, the mode of action, the transcriptome analysis have been accepted by BMC Plant Biology, another journal. Thank you to you, to all for
Thank you, Stefan and Pasquale, for this interesting presentation. And let's move to the next speaker. Is Giuseppe Lima from the University of Molise, and he's talking about an integrated protocol to mitigate olive quick decline syndrome caused by Xylella fastidiosa subspecies pauca in southern Italy.
Thank you, chairs. Good afternoon to everyone. Here I synthesize a multi-year research aimed at set up a field protocol to mitigate symptoms of Xylella fastidiosa on olive in the infected area. We tested different products, and after a longer preliminary screening, we selected 3 most effective products, and these products are commercial fertilizer, commercial formulate of products, in particular Calyx zinc, Calyx copper, and Silifors. The first 2 are phosphites. The characteristics of these products are that they are completely systemic products with phloematic and xylematic movement in the plants. Silifors is a silicon-based product that is reported to reinforce the cell wall and increase resistance to biotic and abiotic stress. In vitro assays evidenced the stronger activity of zinc and copper phosphides, and these products in different tests, in particular in a time-kill contact assay, completely kill Xylella cells, demonstrating then a strong bactericidal activity, eliminating completely within 2-4 hours of exposure the survival of bacterial cells. We moved in greenhouse trials during an year of experiments where we used the most effective products, zinc and copper phosphides, and in these trials, particular Calyx zinc demonstrated good activity, reducing the disease severity by around 50% in respect to the untreated control. Both products reduced also the bacterial population in plants. Then, in field experiments in an olive orchard with mature susceptible plants of Xylella di Nardò, we applied these products within, and this is a very, very important aspect, we used these products within an integrated pest disease management. During the year, we applied 5 treatments using Calyx copper and zinc in alternation, and together other products in a protocol that includes also different fertilizers, and also treatments against the vectors with insecticide, combined also with agronomic practice, as in particular light pruning, yearly light pruning, and soil tillage that, as we know, are very, very important to reduce population of Phalaenus, of the vectors. The results showed that after 3 years of these treatments, the disease index was reduced by 50% around with respect to the control, and also the bacterial concentration in the treated plants were reduced. Also, olive yield and oil quality, as demonstrated by analysis, was improved. We obtained an extra virgin oil with also improved content of polyphenols. Obviously, also the production of olives was improved. In conclusion, the selected fertilizer with strong collateral effect. We don't forget that these are not phytosanitary products, but are fertilizer. And then the activity is a collateral activity against bacteria. Then these products can mitigate, as we demonstrate, symptoms and reduce bacterial load within, while improving also yield and oil quality. The use of these products within a broader integrated pest disease management promote long-term coexistence with the pathogen in the Xylella infected area, At the moment, this is the only possibility we have to contrast this pathogen. Don't exist a product, a resolutive product, and then an integrated approach such as protocol is one of the most of the best available possibility. Furthermore, this protocol can be used also to preserve, to mitigate damage of Xylella on the landscape, on the environment by protecting monumental trees that are widespread in the Salento area. And also is important, don't forget that also tolerant, resistant varieties are not immune, as we evidenced in different meeting. And then we have the need to set up also a protocol such this to contrast not only Xylella, but also the other pathogens that affect the new cultivation, intensive, semi-intensive plant of resistant tolerant varieties. Considering also that these products we selected are polyvalent, other than bacterial activity, as we evidenced in other research, are also have also a stronger antifungal activity. Then in an olive orchard we can use with a unique treatment, we can combat bacteria and also fungi. Thank you very much for your attention, and I would like to thank all the peoples at a different level that contributed to this research, colleagues from, in particular, from the University of Molise, and also from Institute for Sustainable Plant Protection of Bari, and with a particular thanks to our young co-workers, and also olive farmers, peoples from companies we cooperate with us, and all persons cooperate with us in this research. And this research was funding good part with the project Integrolive. For further information, you can also visit our website. And this research was published on plant disease. Thank you very much for your attention.
Thank you, Giuseppe. Now is the time of Luis Alejandro Moldo Santos from XIC that will tell us about the management of olive quickly climb syndrome using synthetic bacterial communities.
So, good afternoon, everyone. I would thank you for the presentation, and also would like to take the opportunity to thank the scientific committee and the organizers for this opportunity. So, we all know Xylella fastidiosa, and that is how it's being managed, which is basically using an integrated disease management approach. A lot of efforts has been performed in the outer layers, so in exclusion, so with the mandatory surveillance, with eradication, the control of the vector, and as we've seen in session 1, the search for tolerant or resistant varieties. But what about the protection and therapy strategies? Sadly, at the moment, there is no efficient treatment to completely cure infected plants. And also, we need to understand the context in which we are, which is basically revolving around the European Green Deal and the Farm to Fork strategy, which is also supported by the Sustainable Development Goals, which the general thing would be that we need to reduce the use of traditional chemical pesticides. So there is a need to find efficient and eco-friendly alternatives. There is a lot of research that is ongoing, which we will see during the conference, of course. Some of them are focused more on chemical control, but are more environmentally friendly, such as peptides, plant-derived compounds, or nanocrystals. And also there are studies about biological control, such as phages. And in this case, we'll be focusing on endophytic bacteria. So what can they do? They can interact directly with the pathogen, interfering with their processes, for example, by producing antimicrobial compounds. They can do quorum sensing and quenching, interfere with that. They can compete for space and resources, and they can also interact with the host. So, for example, they can activate the plant defense response and regulate the plant growth, etc. So why, instead of using one, why don't we mix them together? And this is a little bit the idea of the bacterial synthetic communities, also known as syncoms. They have a lot of advantages, which include higher niche coverage, resilience to perturbations, higher stability, and since it is composed of different individuals, we can have multiple mechanisms of action at the same time. So the objective of this study was to characterize xylem-inhibiting bacteria with promising traits and design syncoms for the biocontrol of vascular diseases, specifically in this case olive quick decline syndrome in olive trees. So we started with a collection of 300 strains that were isolated from olive, and we did a pre-selection of 100 of them. And from there we did different experiments, which I will be quickly explaining, until we designed the syncom. The first one was related to the evaluation of direct and indirect biocontrol mechanisms. Specifically, 18 of them were evaluated, 5 related to indirect mechanisms, and the other to direct mechanisms, such as production of lipases, proteases, etc. And we did that using specific media, as you can see on the images, and we used a semi-quantitative scale. I cannot go into detail, but the general idea is that the presence or absence of specific activities was strain dependent, and based on their profile, we selected 26 strains. These were further tested to evaluate which carbon sources they could use, and also their sensitivity to specific compounds, such as antibiotics, osmotic stress, etc. And we also evaluated the activity against Xylella fastidiosa. Generally speaking, most of the strains could use many of the carbon sources and were quite tolerant to different conditions. And also we were able to identify many of them which were active against Xylella. So from there we selected 12, and we did a dual culture experiment, which basically consists of one versus one situation and creating this matrix that you can see. And most of the interactions were positive or neutral, so this allowed us to select 5 strains, and which we organized in 2 different syncoms. The first one is composed of Bacillus, Novosfingobium, and Variovorax, and the second one is composed of Rhizobium, Metallobacterium, and Novosfingobium. What we then did was to completely be sure that they were compatible, so we added them one by one into the mix. They were compatible, so that's good. And then we evaluated that once they were mixed, their activities were not affected, so they still had the activities that they had when they were alone. We went a step further and we simulated the xylem conditions using microfluidic chambers, but in order to track the strains, we needed to use a conjugation protocol to label them. As you can see on the image, we were able to do so, and what we observed was, here we have an example, which is syncom 1, that all of the strains were able to grow together, and they even interact with each other, forming clusters. So, now we move to the in-plant experiments. Here we have 2 different plant species, a model plant, which is Nicotiana benthamiana, and a target host, which is, in this case, olive. We applied the syncoms, as you can see on the scheme, on the black squares, and after 3 days, we applied Xylella. We inoculated Xylella, both cases using endotherapy, so or directly injecting them into the stem. Depending on the plants, the experiments were more or longer or shorter, but basically we monitor disease severity, shoot system height, population levels by qPCR, leaf physiological parameters using proximal sensors, and fresh weight. So on the left, on the right, you have the graph that of the different parameters, how they were, and the different values. The dashed lines correspond to the plants that were not inoculated with Xylella, but were treated or not, and the bold lines correspond to the ones that were infected with or inoculated with Xylella. On the left, you have some images at a specific time point, and the values for the different parameters for Xylella and how they change depending on the treatment. Basically, as you can see, both symptoms improved. So there was not as negative effects due to the infection. So, for example, there was less disease severity, higher stem height, fresh weight, and chlorophyll content compared to the ones that were only infected with Xylella. And something also that I would like to highlight is that the application of the symptoms did not result in a negative effect on the plant, which is also something that we need to check. In this case, symptom 1 was the one that showed the best results. What about the population levels? In the population levels, what we observed is that, well, we analyzed 3 different areas. We had the inoculation point, and then we analyzed an upward zone and a downward zone. And as you can see, there is less population levels in the treated plants, being Syncon 1, the one with the best results, with reduction in around 3 logs of CFUs per gram of plant. In this case, Syncon 1 was the best again. So now we'll be moving from tobacco to olive, which is the target. The trends were exactly the same. So basically here we have some extra parameters. So we have the number of secondary branches and the total length of those secondary branches summed together. But the trends were more or less the same. The application of the syncom did not have negative effects. And when they were applied, it had the preventive effects. So basically there was higher stem height, higher branching compared to the ones that were not treated. For the population levels, the trends were the same, so there was a reduction. There was not as high population with the treated plants, being Syncon 1 the best one. So Nicotiana is a good model to do these kind of experiments. And we're around 3 logs approximately. Both syncoms, due to the promising results, are being tested on field within the context of the PEXIL project. So we developed and applied the syncoms. ACSBio helped us to produce them, well, optimize their growth and produce them in bioreactors. FertInject provided us with low endotherapy devices. And of course, we had the on-field support of the research groups of the area from Italy and Mallorca. And we applied them in olive and in almond. So we plan to monitor the disease development, the population levels of Xylella, and the population levels of the syncom. So the conclusions of this work is that we characterized 105 strains. The biocontrol-associated traits were strain-dependent. The selected strains showed compatibility. 2 syncoms were designed based on that, and that all strains from the syncoms were able to grow together and retain their activity. Both syncoms showed activity against Xylella in planta and improved overall plant fitness, being syncom 1 the best. And as I mentioned, the experiments are being carried out in Mallorca and in Italy on field. So I would like to thank everyone that was involved directly or indirectly within this work, the funding agencies and the EFSA Young Research Initiative, and of course you for your attention. Thank you.
Thank you, Luis. Let's move to the next speaker, is Tufik Elvearino from Bari, and he will talk from detection to control, integrated aptamer-based diagnostics with FEC and antimicrobial peptide strategies against Xylella fastidiosa.
Okay, thank you. Okay, actually this talk is divided into 2 sections. The first one is regarding aptamers, regarding diagnosis, and the second one is about bacteriophages and the use of antimicrobial peptides for the control of Xylella fastidiosa. This work has been conducted in Siam Bari together with other partners and institutions in Bari. The aim of this work actually was started regarding the diagnosis level is to develop a field deployable device that can detect Xylella fastidiosa in a rapid manner, very fastly and economically and so on. So that's why we have thought about aptamers. Aptamers are short RNAs, molecules, or DNA that can fold into 3-dimensional conformation. And having 3-dimensional conformation, they can lodge within cavities in the cell wall membrane of the bacteria, and that's it. once they do go inside, we can label them or we can integrate them with some elements that or chemicals that can be used actually for diagnosis, and not only, but also for control. So not only for diagnosis. Anyway, this is about aptamers, and the second part is about the use of antimicrobial peptide in order to find or to select some peptides that can inhibit the population of the bacteria inside the infected plant and reducing the pesticide use. And bacteriophages, of course, are small bioagent that can infect bacteria, actually killer of bacteria, and they are highly specific to some strains of bacteria or subspecies. And that's why we were chasing after those bioagents in order to use them for the control of Xylella in infected plants. Regarding the aptamer selection strategy, actually aptamers are very tough to be selected, and that's why we have opted for 4 approaches. The first one is the use of centrifuge based on the high molecule of the bacteria of Xylella in order to separate the library or the aptamers that are unwanted from those that we want to work with. The second approach was the filter-based, because since this bacterium is about 3 micrometer in size, so we have used 0.2 micrometer filter in order to separate these bacteria from the aptamers that we were selecting. The third one is about the use of antibody, given that we do have an antisera for this bacteria. So we went to trap the Xylella on these antibodies, and after we went for the aptamer selection. And the first approach was the one-step fluorescence. Actually, we have labeled the library with efflorescence in order to make it faster, quicker, and without laborious work to see if it works, actually. So to increase a little bit the selectivity and the specificity of these aptamers, actually we went to isolate from different orchards and different affected plants the several bacteria that are called like non-target bacteria, non-Xylella, in order to increase the specificity of these aptamers once used on the sap and the plant, or rather in the insect vector. Anyway, we went to epiphytic and endophytic bacteria isolation, and we challenged the aptamers library with these kind of bacteria that we have selected in order to increase the specificity and the selectivity. By the end, we went through next generation sequencing in order to identify these libraries, because aptamers actually are made of 3 components: the 5' terminus, the 30', and there is an N terminal inside made of 40 nucleotides that are degenerated that can take a conformity. Anyway, to identify their sequence, we went through next generation sequencing by doing 22 libraries and passing from one round to another. So we started with the round 1, round 5, round 10, and 15, and so on, in order to compare the sequence and the enrichment that was happening while we were selecting these aptamers. Anyway, these are what you're seeing actually masked, just for confidentiality, are the 22 candidates of aptamers that we were able to select through next generation sequencing, and we compare all the library together. And we went for study their selection by using qPCR and qPCR, and what you're seeing are the different bands of specificity and concentration by using one bacteria to another. And as a last step, we went to the surface plasmon resonance, which is a technique that can measure the affinity, the ligands between the aptamers and the bacteria itself. What you're seeing here is from 10 aptamers that we went to evaluate, we have only 3 aptamers were chosen as promising candidate, because aptamer 3 and 6 and 8, aptamers 8 in the graph in the middle shows that there is a high selectivity, while aptamer 3 there is a low response, while the aptamer number 6 has showed a good compromise between selectivity and specificity. And based on this aptamer, we are now ongoing to do a kind of integration of this aptamer with some CMOT technology that has been developed by the University of Bari. Another approach regarding the control, so we went to use the bacteria, beneficial bacteria, specifically the Lactococcus lactis that can produce nisin, which is a peptide that's usually used in alimentary, in food. The second one is the use of another lactic acid bacteria, which is Leuconostoc mesenteroides. And the third approach is the bacteriophages. Regarding Nizine, this is the first time that this component or this peptide has been applied on a phytobacteria pathogen. And actually, as you can see from the electron microscope and fluorescence microscope, and also the challenging assays, that this molecule has one function very well in disrupting and in killing or in destructing the Xylella fastidiosa cell. And what you're seeing at the right side is the implanta measures or implanta experiment that we have done, injecting the Nizine inside the small Nicotiana bentamiana, and the result was, I would say, optimal. And to make it brief, this molecule actually could be a good solution or a promising solution or molecule to be used or to take a profit of it for the infection in the field. Regarding the second bacteria or lactic acid bacteria, Leuconostoc mesenteroides, we have seen that in the supernatant of this bacteria there are a lot of toxins, so we went to do some kind of proteomics. And after identifying these kind of toxins, we have found that also by making a whole genome sequencing of this bacteria, we have found that we do have 6 encoding genes that worked very well once they were expressed in vitro, and 4 out of 6 were functionally active by lysing the bacterial cell. And also what you're seeing are implanta experiment that showed also promising therapy or treatment. Regarding bacteriophages, since maybe 3-4 years, we went also chasing bacteriophages for Xylella. We have identified the first one called phage MATE2, and you can see these are the particles and the lytic activity that these bacteriophages can exert against Xylella fastidiosa. While recently we have identified another 2, phage Phi1 and Phi3, belonging to different genera of viruses. They were also shown good efficiency in lysis the Xylella cells. And recently, maybe 2 months ago, we have identified other 2 phages that, as you can see from the electron microscopy observation, are very aggressive because they have completely destruct the bacteria cell. Anyway, I didn't put all the data regarding the experiment that we have done for those because for lacking of time. If you are interested, maybe I can give you the right reference for your knowledge. Finally, all these experiments and trials and reassortments between the use of cocktail of phages and single phages and maybe cocktail of phages and a combination between phages and antimicrobial peptides are ongoing in our experimental fields and screen houses in order to see how they behave in killing the bacteria or decreasing the population of Xylella inside the affected plant. This work has been done through not only, but through also 2 projects from the Ministry of Agriculture, Italian Ministry of Agriculture. One is the One Live Xylella for diagnostic, and the second one is ANCO6 for the development of sustainable approaches for the control of Xylella. These are the workers or the team researchers that have contributed to this work. Thank you so much for listening.
Thank you. Now I invite Emilio Montesinos, which will talk about targeting polysaccharides with synthetic lipopeptides as a control strategy against Xylella fastidiosa.
Good afternoon. I would like to start thanking the organizers for permitting to present this presentation. In fact, this is part of a PhD from a former PhD student that didn't come here. Is a kind of novel orientation in our research line on peptides against Xylella fastidiosa, which is using target-oriented approach. And in this case, using the lipopolysaccharides as a target for these peptides. It doesn't work.
Okay, okay.
No, oh. No.
It's about you.
Oh, okay. Okay, sorry, the time is coming. So for those of you that are not familiar with peptides, there are several sources of natural peptides from many living organisms. An approach more efficient is to prepare analogs from natural ones and construct peptide libraries. And then screening for activities, for example, in the pathogen, several approaches are against the membranes, against the biofilm production, interfering with motility, and recently RNA interference, and many other possibilities. And also induction of plant defense on the plants. We have been working for the past years, mainly in the Bexil project, but also in others. And other researchers have been reported the use in vitro or in planta, depending on the cases, of different peptides. Here there is a list of peptides active against Xylella fastidiosa. In our case, our target was the lipopolysaccharides that are presented in the outer membrane of gram-negative bacteria, like Xylella. You can see here a scheme of the molecules. They have the lipid A, which is embedded in the membrane, the core lipopolysaccharide, and then repeated structure polysaccharides. The lipopolysaccharides in Xylella have been reported as important in the infection process, especially for immune evasion from the host, biofilm formation, and also obviously for membrane integrity and permeability. Here there is in the left hand a list of the lipopeptides we have tested. I'm sorry because the code is one amino acid letter code, which probably you are not familiar. But we used a very simple test for studying the interaction, which is the endotoxin, limulus, polyphemus, ameboside lysate, which is used for lipopolysaccharide testing. So we make it in contact, the lipopolysaccharides, with the peptide, and we see if there is signal or not. If there is not signal, it's because the lipopolysaccharide was quenched by the peptide. So using this approach, you can see here on the left hand a first approach comparing the lipopolysaccharides from E. coli and the Xylella fastidiosa. And the lower data indicate that there is interaction because there are no lipopolysaccharide free in the interaction test. So for example, you can see here several peptides that are active. On the right part of this graph, there are reference peptides like colistin. And in the right hand of the slide, there was a test increasing the concentration of polysaccharide for a standard concentration of the peptide. You can see two peptides here. One is the 178. There is no effect, so no interaction. And the 473, the lipopeptide, there is an effect of quenching, so interaction. But interestingly, this peptide, the best one, the 473, has a dose-response relationship. You can see here in the left hand how viability, using a viability qPCR method, is decreased as soon as the concentration of the peptide increases. So this indicates that the peptides are bactericidal because kill the bacteria. And on the right hand, you see that this effect is not exactly equal for all the subspecies of Xylella. Even it depends on the strain, as you can see here. And interestingly, for example, the Pauca strains are usually less susceptible to, in general, to peptides and other antimicrobial compounds.
You can see here in the left hand how is the interaction using one modality of the BP473, which is linked to carboxyfluorestein. So in the lower panel, you can see by the confocal microscopy how the peptide is interacting with the cells. A not treated control with Xylella, and with the peptide, you can see in the right hand how it is lytic effect on the cells. We use Nicotiana bentamiana as the plant model, applicating the peptide by micro-injection. And the first thing was that this lipopeptide that interact with LPS has no effect, significant effect on induction of genes compared to BP178, which has a strong reaction. Interestingly, injection of the fluorescent formulation indicate that the peptide is going up and down within the xylem and the vascular system of the plant, especially to the leaves from the point of inoculation. And this takes time, but is still active at least for 7 days. And finally, the infection control assays, I'm showing only here, obviously with Nicotiana bentamiana after 65 days post-inoculation, that in the left hand you have severity of symptoms. And for example, in one of the fastidiosa subspecies isolated from Mallorca, it was very effective. And also in the Dono strain, which is a pauca, but not significant in the other two strains used. When compared with the population level, in the case of the IBA strain, it was clear that there is a relationship between disease control and population level, but not for the other cases. So the conclusion is that this kind of lipopeptide and others lipopeptides are very interesting because target directly through the lipopolysaccharide of the Xylella, and then are effective. And this last slide is a proof of concept. We should go to almond, to olive, and to other hosts to verify this effect. I have to tell you that on almond it is working, like in Nicotiana, but then the final aspect is the proof of concept at field level. And we expect, since we have no project ongoing at that time, probably this will be for the future for others. And finally, many thanks for your attention. This is mainly of results from different projects, national and also from the European project Bexil, and that's all for my side.
Thank you, Emilio. Next speaker is Pasquale Benedito. He will talk on the evaluation of thermotherapy protocols on nursery plant material for the management of Xylella fastidiosa.
Well, yes, we made a work about thermotherapy. Our institution, that is the Center for Research, Experimentation and Training in Agriculture, Basile Caramia of Locorotondo, and the Institute for Sustainable Plant Protection of CNR of Bari. So our work is made to, well, there is a problem. Okay. Well, effective sanitation of treatments are essential for the nursery sector of our region to prevent pathogen spread, to ensure safe plant propagation. And so inside the Bexil project, there is an entire work package 3 dedicated to the thermotherapy and its application. Well, it's very important because the objective is to identify thermotherapy conditions able to control Xylella fastidiosa. It is very important for our sector. There is also and the hot water treatments protocol at 50 grades for 45 minutes that is recognized by EFSA for grapevine nurseries. Well, we made different treatments during the period between 2024 and 2026. Well, we made different treatments with hot water temperature in a grapevine nursery farm that is Murciano in Otranto, in the province of Lecce. So we worked on bare root plants, on cuttings, and science for grafting for different kind of species, such as walnut, white mulberry, figs, and sun fruits. So after the treatments, we made several kinds of experiments, and so we potted the bare root plants in controlled conditions. We made the grafting of the science that we made the different treatments, and also the different cuttings of the several species were placed in flags containing water to evaluate the bud burst of the plant propagation material. And we made it in a room with temperature and moisture that was controlled. So we measured different parameters, such as the bud burst of the plant material, and also the graft success rate on the grafted plants. So these are some results. Well, okay. Well, during the 2 years, 2025 and 2026, we had a good response for 2 species at the different temperature that we tested, in particular for walnuts and mulberry. And so while for Ficus carica, and especially for the different stone fruits, such as Prunus dulcis, but also on different rootstocks that we tested, such as GEF 677, we had not good response, especially at the temperature of 50 grades for 45 minutes. Well, for cuttings also, we had a good response on Juglans regia, on walnut, while for Morus alba, only some variety had a good response, especially for the late shooting that. So while for grafted plants, for example, for Prunus dulcis and the Prunus avium, we had a good response for the buds that we grafted, treated at 48 grades for 45 minutes, while for that we treated at 50 grades for 45 minutes, we had a very bad response. Well, so these are the conclusions. So the protocols at 48 grades for 45 minutes showed a good compatibility with the most material that we used, while the protocol at 50 grades for 45 minutes had a bad response for stone fruits and also partial for figs, but for walnut and white mulberry, we had a good response. Also, the CNR have made different treatments on infected material of grapevine infected by Xylella fastidiosa, and almond that infected by Xylella fastidiosa and multiplex. And so we have worked and isolated the material in vitro, and here there is the results, well, we can see that for untreated material, we have the development of colonies, and also for the material treated at 45 grids, while we don't see any colonies at 47 and 50 grids. But in vivo, on planta, we can observe that the treatment at 47 grids that don't show any colonies in vitro, while after the analysis we detected bacterial in plant. While plant material treated at 48 and 50 grids, no colonies recovered in vitro and no bacterial movement to the new leaves in the plant. Also on the almond, we can see that the treatment at 47, 48 and 50 grades for different period of minutes don't show any development in vitro. So thermotherapy is a phytosanitary tool against Xylella fastidiosa. It's very important to extend this kind of treatment on the other deciduous woody plants. In particular, with these results that I showed to you for walnuts and mulberry. But also is a good approach that offers a sustainable, chemical-free strategy to reduce the risk of a long-distance spreading of Xylella fastidiosa. But further research is necessary, is needed to optimize the protocols, to assess the host-specific tolerance and the different behavior between the cultivars, and also validate other heat-based technologies, such as steam, for example. Thank you for attention, and thank you to my colleagues, in particular Pier Federico Lanotte, Maria Saponari, Enza Don Giovanni, and the other colleagues. Thank you.
Thank you, Pasquale, and thank you to all the speakers for keeping the time. Now is the time of the pitch talks, 5 minutes each of you. The first one is Luca Nerva from the CREA, which will present a functional screening of bacterial isolates reveals candidates for the biological control of Xylella fastidiosa subspecies fastidiosa in Greva.
Thank you. So today, just a brief overview of the work that we are doing in the project Novixgen. We are working on grapevine, and specifically in this task we are trying to exploit the biodiversity of grape-associated microbiota, starting from a collection that we built up during the years from previous European project. We have thousands of isolates in this collection, and starting from this one, we started by analyzing plant growth promoting traits to identify isolates that can be used to fortify and implement the response of plants. And then we tested these isolates against Xylella fastidiosa. We started from about 200 isolates at the beginning, and then we reached a number of 100 just because we tested these isolates on the same media on which we grow Xylella. Then here is the result. We identified 129 isolates that are not effective in slowing down the growth of Xylella, whilst 9 resulted very effective in blocking the growth of Xylella directly by impairing the growth or indirectly by producing diffusible molecules into the media. Starting from these 9, we developed a microbial consortia that was applied to grapevine plants at the Balearic Island. We were hosted by some colleagues there. And then we compared plants inoculated with our microbial consortia, a plant untreated, and then infected with Xylella fastidiosa. Here you can see the first results. So looking at the degree of the damage of these plants, you can see that the glass is very reduced in the plants that were inoculated with the microbial consortia, confirming that the isolates that we selected from the in vitro screening are also able to impair the growth of Xylella, or at least to induce some responses in the plants that can reduce the degree of the damage. We obviously analyzed several parameters like the first leaf upper of the infection, the second leaf, the match spatial, and all the parameters of the infection. But at the end, the plant treated with the synthetic communities are more resilient to Xylella fastidiosa. We are now working also on the molecular analysis of these samples, starting from spatial. We decided to analyze the microbial consortia to see if it is there, if we can see any adjustment also in the microbial endophytic community of the plants, and also to look at the transcriptome of the plants to see how they are modifying the gene expression. Thanks to these very nice results, we also started to analyze the same approach in olive. And we also identified several isolates that are effective in reducing the growth of Xylella pauca in vitro, and we are now starting the experiment in plants. And this is all, and thank you for your attention. If you have any question, for sure I'm here.
Let's move to next speaker is Reinaldo Rodriguez de Souza Neto, and he will talk about rational discovery on an FTS targeting, compound inhibiting in vitro growth across Xylella fastidiosa subspecies.
Hi, good afternoon, everyone. Today I'm going to present my results about our study that's rational discovery of FTZ target compound inhibition in vitro growth across Xylella fastidiosa subspecies. So FTZ protein is like filament temperature sensitive Z gene. That's the gene responsible for the cell division and is conserved in all bacteria. So as this is a conserved gene, this became a target for design of new antimicrobial compounds to inhibit the growth of the bacteria. Based on that, the goals of this study was identification of novel potential inhibitor molecules targeting Xylella fastidiosa, FTZ, to control bacterial growth in vitro. So for more details about the methodology, you can check this in the poster, because I have 4 minutes. And so the first our results was to take all the sequence of FTZ proteins from all available genomes of Xylella fastidiosa and make a phylogenetic tree. We also include like Xylella taiwanensis as outgroup. And as you can see here, the proteins are very conserved in all bacteria. Based on that, based on the structure described on the literature and also the library of our colleagues from University of Perugia, we selected 22 analogs to make a screening against Xylella fastidiosa. And based on the results, we selected the compound 18, that was the most potent against the Xylella fastidiosa, and we test this in vitro about the bacterial growth, and you see a dose response effect against Xylella fastidiosa subspecies pauca. And with 200 micromolar of this compound, it was able to inhibit Xylella growth, biofilm, and also CFU. And also we obtained the similar results against subspecies fastidiosa, that with 200 micromolar of this compound was able to inhibit the bacterial growth, biofilm, and also the CFU. And similar results we obtained against the subspecies multiplex, showing this compound has a potential to inhibit the 3 main subspecies of Xylella fastidiosa. Here we also perform a phenotype analysis using the transmission electronic microscope. Here is the control of the bacteria normal, and in 200 micromolar we can see the phenotype described in the literature, that's the elongated cells and also a twist between the cell division, showing that this specific compound is targeting the FTZ as we expected. And we also make a test of phytotoxic acid. We infiltrated this compound in olive leaves, and after 2 months, we didn't see any clear results about the phytotoxicity. So, as partial conclusions, these findings support that FTSZ is conserved and potentially druggable target in Xylella fastidiosa, and identify compound 18 as a promising agrochemical candidate for developing of a novel antibacterial approach against Xylella fastidiosa. For more details about this, we are invited to visit the poster 61, please. So I would like to thank the committee for the opportunity to share the results, and also my colleagues from CNR, from Institute of Sustainable Plant Protection, also for the Institute of Crystallography, and also from University of Perugia. Thank you, everyone.
Thank you, Reinaldo. Now is the time of Philip Vieria, which will talk about the microbial control of Xylella fastidiosa through a novel antagonistic protein from the plant Philosphere.
Okay, thank you very much. Thank you everyone for the opportunity to present the work that I've been developing during my postdoc, which basically sets on two basic principles of ecology, or bacterial ecology if you prefer, which is when a bacterium, in this case invader bacterium, arrives to a new environment, it either adapts to this environment or gets eliminated. That means that bacterial interact with each other, and therefore you can characterize these interactions in order to identify species that can inhibit the growth of a particular bacterium. Of course, in our case, we are interested in Xylella fastidiosa, and Xylella fastidiosa is our invading bacterium, and our established microbial consortia is the Philosphere of Arabidopsis thaliana. And what we settled to do was to characterize the molecular interactions between the Xylella fastidiosa and these different bacteria that compose the microbiome of the leaf of Arabidopsis. So, and that's what we did, and we took a screen-like approach in order to identify specific strains that can inhibit the growth of Xylella fastidiosa. I don't have a lot of time to go through this, but basically we characterized the molecular nature of these inhibitory factors. We identified and got particularly interested in a group of bacterium that belong to Bacteriodota. They are spread across 3 different genus, Chrysobacterium, Flavobacterium, and Atrobacter. And we identified a common protein present in this bacterium. Importantly, we produced this protein and purified this protein from E. coli expressing cells, and shown that a protein produced from E. coli derived from these strains is able to inhibit the growth of Xylella fastidiosa. And taking this approach, we basically designed a synthetic community of these, comprising these Bacteriodota-like species, inoculated plants, and see if they can inhibit the growth of Xylella fastidiosa in planta, which we indeed observed. The name of the protein is for now cell2. This is the predicted protein structure of this protein. And if you want to know what we think it's doing to Xylella fastidiosa, you are invited to go to poster 68. Thank you very much.
Thank you.
Thank you. Let's move to the last speaker. He's Muhammad Amir Muawiga. He will talk on nanotrapping Xylella fastidiosa, morphophysiological, molecular, and hyperspectral evaluations to uncover mechanistic efficacy and disease reduction potential of cellulose nanocrystal endotherapy.
Thank you so much, and good afternoon, everyone. And thank you to the organizers for giving this opportunity to share insights from my PhD. This part of my PhD was conducted in the Institute of Sustainable Agriculture, Sussex, Cordoba, Spain. So basically we all gathered here to discuss this problem, and the problem is the biophil occlusion of the xylem vessels. So, and besides this, I want to stress that this problem is a multi-strain crisis, and not only one host, but many hosts are affected by this. So we propose a solution that the targeted cellulose nanocrystals-based endotherapy can lead to effective cure of this disease. So basically, this is a circular plant protection measure, and these cellulose nanocrystals are derived from the waste pulp of the softwood, and their dimensions is 5 nanometers in width and up to 500 nanometers in the length. And they have a negative surface charge due to the sulfate ester groups, because it is processed through sulfuric acid hydrolysis. So this nanotrapping hypothesis is that these cellulose nanocrystals can physically lysis the bacterial cells without putting them into the viable but non-culturable, that is the dormant state of this bacteria. So during this study, the experimental framework was multi-tier, from in vitro growth kinetics to biofilm to membrane damage towards the endotherapy in Nicotiana benthamiana plants. That is a model host for this, and to see the disease severity reduction and the physiological homeostasis in the through spectral reflectance. So the results were very interesting. Based on the in vitro results, we found that on the CFU-based assay showed a significant reduction of growth as we increased the concentration of cellulose nanocrystals, and the top two tested concentration, that is 0.5% weight by volume and 1%, they significantly reduced the production of CFUs. I forgot to mention that we studied the Pauca Codiro strain and fastidiosa temecula 1 strain. So both the strains were effectively reduced in terms of growth, not only in the terms of CFUs, but also in the terms of turbidimetric growth parameters. Besides this, we observed a hormetic response that initially when we increased the CNC concentration, the biofilm production was increased, but effectively on the top tested concentration, there was seen an obvious decline in the biofilm production. And besides this, based on the cell viability assay, we also found that in comparison to the acute stress, the chronic stress resulted in the more membrane damage, which means that the exposure to the cellulose nanocrystals have more effectivity to suppress the disease symptoms as well. Under implanta conditions, based on the endotherapy, we observed that the bacterial movement was reduced to significantly to different spatial locations of the stem, and the AUDPC also proved that in Codiro, the disease symptoms were reduced up to 80%, and in Temecula up to 60%. Also, based on the spectral reflectance, we observed that there is a clear rescue method observed. So, lack of time, I would like to continue this conversation on the poster session. My poster is 56. Hope to see you there. Thank you so much.
Thank you. We are perfectly on time, so we can start with the session of questions and answers that I expect. All the speakers are... Se manca lui. All the speakers should be there, but they are not there. Who is lucky? Pasquale. Okay, sorry. Thank you. So the session is open, and I expect a lot of questions because it's not subject control. So feel free, start.
Yeah. So I go first then. I didn't prepare to go first for any question, but this time I didn't see any raised hands, so I do have general question for all the talk that actually using chemical compound as antimicrobial treatment to control Xylella fastidiosa, right? We all know that antimicrobial resistance, it comes when you use antimicrobial compound to treat the bacteria, but also select the resistant bacteria. So during all of your treatment, it will be in the talk number 2 4, 5, and maybe number 8 too. If you have find any resistant strain to the compound that you used in your trial.
So as far as I understand, if we have identified, someone of the speaker have identified resistant strain to some of the chemical that... No, first I would say that according also to EFSA, there are no direct chemical control in the plants able to eradicate the Xylella. So I don't know the other speakers.
Yes, well, I have some answer. It is highly dependent on the mechanism of action of the antimicrobial, as is the case for other compounds. For example, if in the case of peptides, if the mechanism of action is multi-action, for example, affecting different components of the membrane of the bacteria, it is not expected to have resistance. If the mechanism of action is interfering with one step in a pathway, in a biochemical pathway, the probability is higher. So it's a question of this case. But there are different mechanisms of resistance. For example, quenching the compound by producing exomucopolysaccharides, which is one of the processes. Other is degrading the antimicrobial, degradation of the antimicrobial by producing some enzymes. So it depends very much on the mechanism. But my opinion is that we should not rely in only one compound and one mechanism of action. So we should use combinations. As in the case of syncoms with microorganisms, we have to do that. But this should be tested in the real conditions. And we have not yet done that, at least with functional peptides.
Yeah, for the functional peptide, for example, in the case of colistin, when using as antibiotics compound, right? It targets the membranes. That means it can target some gene in there,
yeah, I know the case of colistin, because the case of colistin...
Economicin in there when testing, it actually not on the membrane, it's on the proteins and DNA synthesis. So you are right that the target or the mode of action of all the compounds are different. But we always underestimate bacteria in terms of they are simple, but they are so quick now. Xylella consider slow grow bacteria, but compare for the, but they do have the 3-day or 4-day of doubling time. That is why we worry that because they're slow, so they can develop resistance when they are slow growing. We experience the problem with tuberculosis in the humans. So I don't know if we will, as a plant doctor in this case, do we need to deal with that in the future when we totally now ignore that side effect of the using antimicrobial compound on Xylella fastidiosa?
Yes, but antimicrobial resistance that pose a risk to humans is mainly based on plasmid-borne resistant. If these plasmids can be transferred to human bacterial pathogens. That is not the case in many antimicrobials. You know that in Europe, antibiotics are not allowed to be used. For example, kanamycin is not allowed, kasugamycin is not allowed, streptomycin not. So we have not this risk. This is the reason for what it's not authorized. They are used in agriculture.
I feel like we will consider talking like that forever, but because you say about the horizontal gene transfer for antibiotic resistance, yes, that is one mechanism, but it also vertical gene transfer. Tuberculosis, for example, they don't really rely on plasmid to transfer the resistance, but they can generated resistance, like in the housekeeping genes, like Zy and PASI. So this can also happen with Xylella. We never analyze if Xylella have how many plasmid and copy number plasmid, we don't know. And so far, it seems like they are quite fit when they find the right host. So I really don't know. But I just think that it would consider if we can find the exact mechanisms of the chemical compound we use on the bacteria, where it acts on the bacteria, on membrane, on DNA synthesis, or RNA synthesis. So that is now apply that. If we know like that, we can see the dose we use, the treatment time we use to eliminate the potential development of the resistant strain in this case. But I'm happy to talk about that after this conversation, because if not, we can talk further. By the chance, I am the antibiotic resistant researcher in animal health and human health before I come to plant health 5 years ago. That is why I have the concern, because I see how quick the resistance evolves since I started 15 years ago and now. Thank you.
Okay, thank you.
I have a question on the thermotherapy. I think it's very good results. So we finally have some results in species different from grapes. So we have almond, other species, malus. But the question is, what is the perspective applied this technique also to woody ornamental, for example? Because I think sometimes it's interesting to know if there is some easy treatment for cutting some other plants on woody ornamentals. So what do you think?
Yes, we see that thermotherapy was a good alternative a treatment for deciduous woody plants. And so it's very practitioners, and it's not difficult to do. It's necessary to have the right machines, and so where we can, and some nurseries would have this kind of condition, because they can control in every moment the right temperature that the water is inside, and so on. And so it's interesting to test these techniques also to different varieties, different species, to verify the behavior of the different plants, of the different kind of species and varieties. And so it's important in the future you can extend the official protocol that exists now for grapevine also on the other kind of species.
Okay. Other questions? No. I don't know. Yes, it is. Okay.
Thank you. This is a question for the second speaker. My name is Blonda from CNR. I think that there is a convergence of evidence between the results obtained in this study according to the use of copper and zinc bioproducts and the study carried out by CREA by using the Dentamate products, which include also citric acid. So my question is, have you studied the doses of your treatment depending on specific cultivar and age of cultivar? Or the dose is the same for all of cultivar and age of cultivar?
Thank you. Thank you for your question. In our case, we used two products, copper and then zinc phosphides, that are known to be very highly systemic products that go both in the xylem and in the phloem system, and then have a complete system. In the case of your product, I don't know if the copper citrate used is completely systemic. I think that go only on the xylem. Concerning the age of plants, we obtained, as you can see in my presentation, we used also young plants, pot ground plants in greenhouse, and also mature plants. And in other field experiments we have in different orchards, we are obtaining the same results. Okay. And another important aspect is also the mechanism of action. That is a combined mechanism of action. These products have a direct action on the pathogen, a strong antibacterial activity, and also an activity on the plant, since they are resistance inducers.
Okay. There is a question down.
Hello, even if you don't see me. This question is for Muhammad Amir Muaya. Sorry for the pronunciation, I don't know. Okay, hello. This is just a curiosity actually about nanotrapping, but it's, the action of these nanoparticles is just to inhibit the initial adhesion of the bacterium, or it has, I don't know, any effects on biofilm that are mature, if my question is clear.
Thank you so much for your question. If I can understand well, you want to know about the insights about the biofilm production, and if this is the first study, or do you mean that this is the first study to read, or could you please elaborate?
Yeah. No, I was just wondering if these nanoparticles are actually have any effect on biofilm that are already existing and mature in a certain way, or they just block the biofilm, the production exactly of the biofilm. I don't know if there is a difference,
Yeah, now I get it. So
Thank you.
Instead, in addition to just reducing the biofilm production at optimal dose, we also studied the cellular viability through PMA-XX dye assay, the mechanism for which is that the compromised membranes can be identified through viability cell assay. So it is also proven in this study that when we increase the exposure time, it damages the envelope of the Xylella fastidiosa. So in addition to biofilm reduction, it also damages the physical membrane, and based on some previous studies that are done by my colleagues, we understand in depth that cellulose nanocrystals physically damage the envelope, and which reduces the growth, overall growth of the bacterial population. And as we all know that when the growth is reduced and the population is reduced, it is not able to create a biofilm production. It doesn't go into the biofilm production. So that's why it's a potential curative solution. Thank you.
Thank you. Other questions? Then, waiting for another question, I have a question for Luca Nerva. If I was not wrong, you first select your bacteria, endophytes, by their plant growth promoter activity and then for their biocontrol.
Yes.
So it was a sequential.
Yes, exactly.
Why this, not the opposite?
Just because we isolated these bacteria as endophytes in general from wild grape, not really wild grape plants, but grape from vineyards. So we don't know which bacteria can be beneficial or detrimental for the plants. And so the first screening was to identify the one that can give something to the plants from helping absorbing any kind of nutrients or anything else. So it was a first step just to understand if they can do something good for the plant. And from there, we decided then to screen, because most of the endophytes that we isolated have plant growth promoting traits. And this is why we first started from there, because we know that they can provide both effects. So beneficial from the biocontrol point of view, but also beneficial for the plant growth. And we think it's very important, since the protection from infection, or at least reduce the symptoms that the plants show when they are infected. It's also a matter of how the fitness of the plant is. So improving the plant fitness using these kind of plant growth promoting traits, we think it's important.
Question here.
Thank you. PSJN is being tested now in olives by Biovexo, but it was already tested also in grapevine in California with good results. So what is the plan now that we have outbreaks of Pierce disease in Puglia, in Mallorca, to test also PSGN on grapevine?
Yeah, good question, thanks. We need another project for that. Sorry, we are depending on grants, and but yes, sure, we should try that in Mallorca and also in Puglia on different cultivar. Yeah.
Thank you for this question, because I forgot to tell that these products from Biovexo, these are ready for the use. So the PSJN preparation is a dried preparation that is produced by ACSBio, one of the company. I do promotion, sorry for this. And the other product, Sankari, the insecticide, is already in production. On grapevine, it will be tested. It will be tested because there is a project that is just starting. I think, and we in Bari were very interested in promoting this use, because the leaf of the grapevine is another leaf. It's not olives. So there are also preliminary evidence from California, as you said, so it will be tested.
I have a question to Luis Moll, that your SYNCOM one that reduced the population level of Xylella in Nicotiana ventamiana and also in olive plants. So do you think that the same SYNCOM can be effective in different plants? Is this possible, or it have to be designed for each kind of plant?
Thank you very much for the question. Ideally speaking, I would say that if possible, SYNCOMs should be designed for the, so for example, if we isolate from almond, from bacterial isolates, so probably they will perform better in almond because they are more suitable in the conditions of the sap composition. But
that does not deny the possibility that this could actually happen, that, for example, the syncom that we design could work in other crops. But it's something that should be tested, and we cannot generalize, because it could be that maybe one of the strains does not perform well in another crop. So we would have to test that. In order to do that, we should monitor the strains population in the plant, which is something that we are working on right now. We are developing qPCR specific for a strain, and we will see how the populations develop in the Nicotiana experiment and in the olive experiment, since we have the DNA from those. So yeah, thank you.
Thank you. We have, okay. Grazie, Antonio.
So thank you. Maybe just this wrap-up question for this session. And it's a question for all the colleagues, speakers this afternoon working on biological control, because it seems that the Commission is going to change the current regulation for registering biological control agents. So I would like to know your perspective from the specific case of Xylella and the biological control agents that you are developing. I mean, will it be more readily available in the near future, or there are some particularities that need to be taken into account? Or?
It's a very good question, because it takes a lot of time to register biocontrol agent, and also it costs a lot. And even when we show some promising solution with slide, I'm even not sure some of them will go to the market, because it can take 4, 5, 6, 8, 10 years, and to have appropriate resources for that. And you see also the fact that these microbes, they have also biostimulant activity, but we cannot say the claim to the farmer to use that to reduce the Xylella, even with the biofertilizer, and now we see that. But we need to register as biocontrol, and it's a very long story, except if the companies, they have a lot of money to invest for that. Yeah. If there is a change in the regulation to go faster, of course, to be still safe as before, this will be better for that. But this is in the end of Commission and EFSA. Yeah.
Yes, I want to comment something that was discussed in the Vexil project meeting yesterday. Well, I think you are referring to biocontrol, that is an example, microorganisms, for example, as plant protection products. If this is the question, I know this is good, because probably most of the microorganisms will be in a faster way of registration, probably. But the question is that what happens with other biocontrol systems that now are even commercialized as biostimulants, in spite that the EU regulation on biostimulants is very clear, which is only for abiotic stress. But also here in this session, there has been some comments saying this is a biostimulant, but the effect is directly targeting the pathogen. So this is not a biostimulant under the regulation, but depends on the country in Europe, because the European regulation and the national regulations are still coexisting. So you can registrate something that is not according to the EU regulation. And I think that these regulations should be clearly depicted from the EU Commission. And because if not, we will see that a microorganism is registered as a plant protection product, but also is registered as a biostimulant or even as a biofertilizer. But it is well known that the effect is not biostimulant only, maybe, but also is targeting the pathogen or the pest. So I think this is a question that within the EFSA needs a strong discussion to prevent illegal marketing and commercialization of products.
Thank you.
I think EFSA has been called.
Verify that EFSA does not make legislation, so we only assess the risk, but following the legislation made by the European Parliament and the implementation by the Commission. So EFSA is not deciding on the legislation, it's the European Parliament is deciding. The colleague from
Hello, everybody. I am a little bit hidden here. I'm Karin Instedt from the European Commission. Well, I just wanted to echo what Giuseppe said. I mean, EFSA is the risk assessor in Europe, and risk management is the Commission together with member states. So EFSA is doing a great job to compile the information and to do the risk assessment, but when it's come to taking regulatory decision, EFSA is lucky not to take this decision. That's up to us and the member states. As regards the new regulations, that's also not the Commission deciding. The Commission is doing a proposal, and then this is decided by the member states and the Council and the Parliament. And the Commission, I mean, is involved in helping with the negotiations, but it's decided by the Parliament and the Council. I think that's also important to recall. And yes, I mean, as regards clarity of the regulations, I see your point. It's sometimes very difficult because it's very complex, and it's particularly complex when it's coming to innovations, to innovative products, because of course the regulation, the legislation is in a way always behind the new developments, so we need to adapt, and we need to adapt quickly and be flexible. But if there are good new ideas coming up, then maybe there is a need to have a talk with member states, rapporteur member states, regulators on what is the best way forward to get these to the market. But also regulators don't have the crystal ball to know what is coming. So there is a good communication needed there. Thank you.
No? Okay, it seems that all questions were satisfied. And we close the session exactly in time and go for the coffee. Thank you to all.
Thank you.
It doesn't work. No. No. Yeah, it's working. It's working.
Yeah. Hello. Okay. All right, let's sit down so we can get started. Now it's working. There are people outside. Should we wait for the people? Or? All right, people outside. 9 minutes late.
Italian time.
All right. We wait another minute until people comes in and we can get started. This is always open. So if I cough or something, it will be amplified.
Yeah.
You just open it? Okay. We don't have water, nothing for the speakers. Very poor service. Okay, now more people are coming in. I see Blanca outside. Emilio is outside. Everybody's outside. We can go on top of the table there. All right. Oh, this guy is working here. Okay, shall we get going? Shall we start? Okay. All right, we're going to get started, so please sit down.
Sit down.
I'm going to start shouting. Want me to say something or you want to introduce me? Hello, everybody. Thank you for coming back for the last session of the day before the poster session. Now we're going to have the session on advances on biology and control of Xylella fastidiosa. So you already met Stefan Compan, was going to be the co-chair with me, and I'm Leonardo de la Fuente from Howard University, USA. So we're going to have a very interesting series of talks on both the biology and as well as the control. So the first talk is by Lena Picenti from UC Leuven in Belgium. And she's going to tell us about the silent endophytome as a driver of disease outcomes in Salicaceae infected by Xylella fastidiosa and Brenneria salicis. Thank you.
All right, so thank you everybody for the opportunity to speak at this conference. This presentation will develop research done at UC Leuven during my PhD thesis, and as Leonardo said, it will talk about endophytome of Salicaceae infected by Xylella or Brenneria. The risk posed by Xylem pathogen in Northern Europe should not be underestimated. If it is well known that Xylella fastidiosa causes major issues on plants such as almond, grapevine, olive, or coffee, in region with temperate climate, question remains for Northern European regions which traditionally have cooler climates. However, several studies have already shown that the bacteria was able to grow even at lower temperature, as we have seen in regions such as Northern USA or Canada. So those regions could become more suitable for the establishment and the propagation of the bacterium. Brenneria salicis causes issues in regions with temperate and mild climate on plants such as willows and cause a disease called the watermark disease. Both these pathogens have something in common. Salicaceae are efficient hosts. But why are they efficient hosts? Because they are common in the riparian tree layers, which have been established as important areas for the disease, because they form a closed mesh network, meaning that if an infection would occur, it would spread rapidly. Salicaceae are also the host of xylem sap-feeding insects, which might be potential vectors. For Xylella fastidiosa, this has been determined through a mark release recapture experiment, through flight meal experiment to see the dispersion capacity, and through a transmission test If you want more information about those tests, you can scan the QR code. Both bacterium are also able to multiply and disperse in the xylem vessels, and this has been revealed through mechanical inoculation. So if you want more information about that, you can scan the second QR code. And the last thing they shared is that when Salicacea are infected either with Brenneria or Xylella, they can be symptomatic or asymptomatic. One hypothesis that we could say about this duality in symptomaticity is that xylem endophyte might play a role in mitigating symptoms development in host plants once they are infected. So the idea would be to first define a core xylem endophytome for Salicacea, then to see the interaction between those xylem endophyte and the two phytopathogenic bacteria, and then to see the potential benefits of targeted co-inoculation of those beneficial endophytes. So we begin with the first definition of the endophytome of Salicacea through culture-dependent method. And we find that 5 bacterial species are shared by all the Salicacea that are tested. So they are Bacillus proteolyticus, Subtilis, Techilensis, Toyanensis, and Velezensis. We can also define a xylem core endophytome for Populus species, which shares 9 bacterial species, and for Salic species, which share 6 bacterial species. Then we wanted to confront this first view of what can be found in the xylem endophytome of Salicacea with some culture-independent method. So we sequenced the V1, V3, and V3, V4 hypervariable region, and if we see some divergence in the genus that are isolated or sequenced, we can see here in green that some of the genus are found in both of the method. And now that we have a fuller complementary picture of what can be found in the xylem of Salicacea, we wanted to select the strain of interest. So we went ahead and did some in vitro confrontation with both pathogens. So here you can see the control plates, and then on the right you can see the plate with an endophyte inhibiting the growth of the pathogen. This allowed us to select 40 strains, and through those 40 strains we did some additional in vitro testing for other beneficial traits such as phosphate solubilization, siderophore production, motility, indole production, biofilm formation, and inhibition of some vascular fungi that can be found in the xylem vessel of Salicacea. And this allowed us to select 15 strains. Those 15 strains were genome sequenced, and then 5 were transformed to express fluorescent protein, which allowed us to track them in planta. So 5 strains were selected for the in vivo inoculation, and they were Erwinia rapontici, Bacillus velezensis, Pseudomonas coleopterorum, Pseudomonas graminis, and the Paraborchilderia phytoferments that Stéphane Compon mentioned earlier. And so during the in vivo inoculation, we of course monitor the symptoms. We check during the inoculation the propagation of the bacteria with PCR detection on the leaf, and we checked for the bacterial load at the end of TSA with qPCR. We also wanted to check with confocal microscopy the localization of the bacteria in the xylem vessels, and we checked the viability of the bacteria with isolation on specific medium. So here's the result for the symptoms. So we did the assay on tobacco and on willow, and we established a visual scale ranging from more or less asymptomatic plants to clearly stunted and wilted individuals. And we observed the plant during 1 month, and then in another assay during 3 months. So on each slide, we'll see framed in blue the plants that were the control, then in orange the plants that were only inoculated with an endophyte, in green only with a pathogen, and in yellowish color with a combination of one endophyte and one pathogen. And so here we can clearly see that we have a mitigation of visual symptom severity in plant co-inoculated with any endophyte across all assays. We also checked the plant height for the tobacco and for the willow assay. After 1 month, we did not see any significant difference. But after 3 months, if we take our color back, we can see that we have a height restoration of co-inoculated plants with any endophyte in tobacco after 3 months. Then we wanted to check the localization in the xylem vessels, and we did not co-localize any endophyte with the pathogen in the vessels, but we did see them separately in different vessels. We also checked the viability. And then we wanted to see the bacterial load. And so this is for the tobacco assay, above for 1 month assay and below for the 3 months assay. And so we can see that we have a lower concentration of Xylella when co-inoculated with all on the fight after 1 month, and with all on the fight except with the Paraburcolderia after 3 months. For Brenneria, we have a lower concentration after 1 month with 2 on the fight, and with all on the fight after 3 months. So in most cases, we have a reduction of the pathogenic bacterial load. Then we also checked for the Willow assay, and here, after 1 month, we did not see any significant difference in pathogenic load, but after 3 months, Brenneria sedicis was lower in the control than with any on the fight. So to sum up, we have a new description of Salicaceae xylem endophytome with 440 bacteria isolated from 43 genera and 104 species. We have sequenced 158 genera and 223 species. From the bacteria that were isolated, we selected 40 bacteria through in vitro confrontation, and then 15 were chosen from in vitro tests for genome sequencing, and then 5 were used as new tool with a fluorescent strain to track implanta. And we can say that we did see an effect of co-inoculation on symptoms development and sometime on a pathogen load, especially in tobacco after 1 month. What would be next is to use metagenomic to better define the Salicaceae endophytome, to use other Salicaceae species for the co-inoculation assay, to maybe use synthetic communities and not single inoculant, and to use a vector to disseminate the synthetic communities. So with that, I would like to thank you for your attention and thank everyone who participated in the project.
Thank you very much, Elena. The next speaker is Leonardo, the co-chair of this session.
Thank you, Stefan. Can you hear me okay? All right, let's see if the presentation. All right, so I'm going to talk about a problem that is not a problem in Europe now. So I'll just tell you what's going on in the US, but it's mainly I'm going to focus on blueberries and the problem with Xylella. The work I'm going to present was done by my students, mainly Mamata and Navdeep. So bacterial leaf scorch in blueberry was first reported in Georgia in 2009, and it's been a problem in the southern states, mainly in the US. Because of these low-chill blueberry varieties, these southern high bush and rabbit eyes varieties, very popular in the south, they are adapted to warmer temperatures. Historically was more cold adapted varieties in the north of the country. I marked the southeast where these varieties are very popular. In the map, for instance, you see in Spain is the #1 producer of blueberry right now in Europe, and mainly in the area of Huelva, which is also like the temperature is warmer, and they're using these varieties that are also being adapted now in China, where the production is surpassing now US, that used to be the #1 producer in the US. So when you have, of course, the expansion of these varieties, there's new problems, and I'm going to tell you what we've been working on blueberries and also in the grapevine pathosystem. We'll be doing some implanta analysis and also some in vitro tests. First, I'll tell you about the blueberry pathosystem. So as part of the project with the Bexil, we tested a set of 31 strains, mainly from Europe, 17 of them from Europe, 3 were intercepted here in Europe, another 1 from the US. And we did greenhouse experiments to assess for virulence. And if you look at the AUDPC, that was the growth curve, the disease progress curve. I mark the ones with the blueberries are strains isolated from blueberries, and the ones with the EU flag are all the strains that we tested from the EU. And you can see there's a big variety from non-symptomatic to severely symptomatic, like this one that it was pretty much one of the most symptomatic strains from blueberry. So this is in terms of the population. So we check the population of the bacteria and the plant, and you see, I mean, there's a big spread of colonization of the plants. All right. So when you see, basically, we saw that also species that we tested in blueberry, they were all causing symptoms, and one interesting one was this strain from lavender, isolated from Ibiza, Spain, that is a Pauca strain. So in the past, there was reports already on fastidiosa multiplex strains, and now we know that also Sandai and Pauca also can cause symptoms and colonize blueberries. So we wanted to complement this with some in vitro studies, and we decided to work with blueberry sap. That was more difficult than we anticipated. This is my student, Mamata. She found this protocol to extract the sap from blueberry. She got to use 130 plants, and she would get 1 ml per plant, 1 per 4 ml. So it was a long work, but she was able to collect some sap to do some experiments in the lab. So we wanted to check how these bacteria, different strains that can cause or not symptoms in blueberry, they can cause also, they can grow in the sap extracted from blueberry. So the first one that you see here, for instance, is we use first a susceptible cultivar called Rebel, one of these low chill varieties. And what you see here in the orange bar is growth in the normal media, synthetic media, PD3, and then the same media with 25% added sap from blueberries. And again, they are separated by the subspecies. So you see that in some cases, addition of the sap will increase the formation of biofilm, and in some cases will decrease, disregarding of the subspecies classification. So at the beginning, we couldn't tell what was the pattern there. And then we, okay, with the multiplex, let's see if we can move forward. Okay, no. Okay, this is in the fastidiosa, the same thing. Some of them will increase the biofilm, and some of them will decrease. Okay. Sorry for the... So basically, when we look at the information that we collected from all these strains, and if I can show it, okay. We saw there was a correlation of the ones that they were making more biofilm when we had the sap, they were causing more severe symptoms, and the opposite, when they were making less biofilm when we had the sap, they were mostly asymptomatic, with exceptions. So I put it on the right there, the exceptions of the strains that didn't follow that, that rule, if you will. So about the 85% of the strains follow that idea, but it's not all of them. So it seems to be not true for everything. But also we did the same thing with grapes, and the same kind of approach. We also selected strains from to testing grapes that were isolated from grapes, one that were isolated from blueberries, and one also were isolated from almonds, both multiplex and fastidiosa strains. And we did greenhouse experiments. And we see here that basically in that case, we saw more a separation of subspecies. So the species fastidiosa, either from almond or grapes, they were causing symptoms, and they were causing also, they were colonizing the plants, while the multiplex not. In general, they were less they were not causing symptoms or colonizing grapes. And in terms of sap of grape, it's much easier to collect, so we can have bigger volumes. We do this at the end of the winter, until the dormant time, and then we can collect a lot, so we can do more experiments with more concentrations. And we use also this set of strains, and we found similar differences that sometimes, in this case, you see the PD3s on the first bar, and then the other ones are 25, 50, and 75% of the sap of grapes. And there was also like a tendency that some of them were increasing, while some of them were decreasing, and some of them were not changing, depending on how we add the sap. And if I can move these, all right. All right. Okay, now it seems to be working better. Not so much. I think it's not working. Okay, now we go. So the almost isolated strain, the same thing we saw. Apologize for the... Okay, that is a difference in the response to the sap. And to summarize that, no. Okay. See if we can, okay. One of them, you see the tendency was like also increasing in some of the multiplex. And when we look at the symptoms in grapevines and how they grow in sap from grapevines, similarly to what I showed you with blueberries, in general, the ones with the high biofilm have severe symptoms. With the low biofilm, some of them still have severe symptoms. And in the case of multiplex, the same thing. The first one follows our idea that the biofilm is correlated to symptomatology, but some of them not. So in this case, the following the rule was just like 70% of the strains, sorry. And we tried with susceptible vector-resistant sap because we wanted to see if they behave differently. And in the terms of blueberry sap, we have a variety called farthing that is pretty tolerant to Xylella. And in this case, we used 3 different strains from blueberries. And in every case, they were producing less biofilm growing in resistant sap. And similarly, when we use grape sap, we also, we saw the same tendency. Using 3 strains from grapevines, when you put sap from a resistant variety, in this Blanc du Bois, it was reducing the production of biofilm. And the last experiment that we did was a passage experiment. So we have the idea that if a strain is virulent and is adapted to sap, if you make serial passages, will be more adapted than other ones. So we marked the strains with antibiotic resistance, and we did like 3 serial passages of 7 days each with susceptible variety in grapevine because we have more. And what you see here is like the strain on the top is virulent, and the strain at the bottom row is avirulent. And each line is like 7 days passages. So you see basically what we saw. I'm going to explain that part. The strain that is virulent by the 3rd passage is adapted to grow much better in sap. You see the increase very much at the end, while the one that is avirulent pretty much died. Even if at the beginning, if you see the first line, at the beginning they seem to be doing very well. So they crash and burn. They start growing very well, but then when you start passing them, they cannot adapt anymore. While the virulent keep going and growing more. So I'm out of time, but hopefully the conclusion is like a lot of the European strains that we tested can cause symptoms and affect these low chill blueberry varieties. We saw a correlation between the sap that we add to the media and the biofilm formation with the symptomatology. And we see that the tolerant sap, the strains usually they reduce the biofilm formation. So we think that is something, of course, in the sap composition that is determining this resistant or tolerance to the infection. So with that, I just want to say thank you for the funding, for Bexil and Bart, part of the people who participated in this research, and thank you very much for your attention. All right. And next, we have Victoria da Silva from Campinas, São Paulo, from Brazil. She's going to talk about the RPFF-mediated DSF production induces priming responses in transgenic citrus sinensis.
So first of all, I'd just like to thank the organizing committee for the selection of our work to perform this presentation. And that said, let's get started. So Brazil is the world's largest producer of sweet orange and accounts for almost 80% of all orange juice consumed around the world. But besides its relevance, the citriculture is affected by several biotic stresses, including the citrus variegated chlorosis, the CVC caused by Xylella fastidiosa. So this disease is reported in several Brazilian states that goes from Amazon to the south of the country. But the problem with this bacteria is that Xylella is a multi-host pathogen and can also infect some other economically important crops in Brazil, such as plum, coffee, and also olive. So Xylella is a pathogen that colonizes the xylem of the plants, and the disease severity is closely associated with the bacteria movement through the xylem. And this movement is controlled by the bacteria coron sensing, which in turn is regulated by diffusible signal factor molecules, which we call simply as DSFs. The DSFs are synthesized by the RPFF enzyme, which is encoded by the RPFF gene. These molecules are conserved short-chain fatty acids responsible for the control of the bacteria gene expression accordingly to the population density. So as the bacteria population density increases, there's an accumulation of DSF molecules that are perceived by the bacteria and that in turn regulate the gene expression, inducing genes that encode for the biofilm formation of the bacteria, and that is the sexual form of the pathogen, and repressing genes that encode for the bacteria movement, and that is the planktonic form of the pathogen. So thinking about this system was asked if plants overexpressing the RPFF gene could interfere with the bacteria signaling, preventing the bacteria movement through the xylem. This strategy was first employed by Dr. Stephen Lindown from UC Berkeley, who generated transgenic grapevines overexpressing the RPFF gene, and a few years later, our research group developed transgenic citrus plants overexpressing the RPFF gene. And what we saw here is that the bacteria that infect those plants show an early increase on the expression of genes that confer an adhesiveness to the pathogen and a decrease on the expression of genes related to the bacteria movement. So when the bacteria infect those plants, they get confused, and that's why this strategy was called the pathogen confusing strategy by Dr. Stephen Lindown. And as the years were passing by, several studies related to DSF molecules were rising. And what we saw is that the DSF not only mediates the bacteria-bacteria communication, but can also mediate inter-kingdom communications. And among many functions, I would like to highlight the role of the DSF in triggering the plant immunity response. So this was the first article that we saw, and here they show by doing infiltration assays into Nicotiana benthamiana leaves with the DSF from Xanthomonas, they showed that the plant can actually perceive those molecules and trigger a hypersensitive reaction response, which is a kind of a plant defense mechanism. In these articles, the authors also showed that the DSF primes the plant immunity system by promoting the jasmonic acid signaling in Arabidopsis and also Brassica. So as we saw this in the literature, now another question was raised, and now we want to know if the DSF molecules produced by the transgenic plants could also be acting as Lister molecules. And to answer that question, the first thing that we did was a hypersensitive reaction assay to determine whether the plants can recognize the DSF molecules. And for that, we infiltrated Nicotiana benthamiana leaves with hydrophobic compounds extracted from transgenic and non-transgenic plants, and we also infiltrated them with synthetic DSF from Xylella. These leaves were detached and photographed under white and UV light, and here you can see the results. So in the regions that we infiltrated the non-transgenic plant extract, there were no necrosis, no AGR-like symptoms. But in the regions that we infiltrated the transgenic plant extract and the synthetic DSF from Xylella, there were necrosis of the tissue, which is correlated with an AGR-like symptoms. So here we show that the plant can actually perceive those molecules, and now we want to know if these plants can, if the DSF can actually elicit the plant immunity system, and for this we did a transcriptome analysis, and we detect 105 upregulated genes, and saw that they were related to the MAPK signaling pathway, the plant hormone signal transduction, and also the linoleic acid metabolism. And when we get deeper into the analysis, we also saw that those genes are related to oxidative stress response, lipid metabolism, ethylene and GI responsive transcription factors, and we also saw the induction of PR proteins. But today for this presentation, I would just like to highlight the lipid metabolism pathway, especially these two genes, the HPL for hydroperoxidase and the LOX2 for the lipoxygenase 2. So the LOX2 enzyme uses linoleic acid to produce fatty acid hydroperoxidase, which can be used by HPL to produce volatile oxylipins, which are important molecules for the plant defense signaling. And these molecules molecules can also be used by AOS to produce JI itself. And the reason why I'm showing you this is because it was already shown that the oxylipine profile is actually really important for the plant defense against Xylella. And actually, in this article, the author showed that the LOX2 pathway is important for the plant defense against this pathogen. So by using LOX2 Arabidopsis mutant, the author demonstrated that the reduction in the 13-oxylipine profile, which is derived from the LOX2 pathway, allows the pathogen to move and to colonize the host to a great extent, showing the importance of this pathway in the plant-pathogen interaction. So the last analysis that I'm going to show you is the lipidome analysis that we conduct to identify the profile of the fatty acids in the transgenic plants regarding the non-transgenic ones. And here, I'd just like you to remember that the RPFF is an enzyme involved in the fatty acid biosynthesis, and it is overexpressed in the plant. So here we saw an alteration in the lipid profile between the transgenic and non-transgenic plants. And what was interesting is that we detect some exclusively fatty acids in the transgenic plants that may be derived from the RPFF activity, since the same substrate used by the RPFF in the bacteria to produce DSF is also present in the plants. We also saw an alteration in the lipid profile between the transgenic and non-transgenic plants. And the lipid that calls our attention was the LPE for lysophosphatidylethanoamine. And the reason why it called our attention is because the LPE was already shown to prime the plant immunity system by promoting JI signaling in rosohomeostasis. So here, what I show you is that the transgenic plants are indeed more resistant to the CVC due to the pathogen confusion strategy. But here, we also saw that the DSF produced by the transgenic plant could also prime the plant immunity system, especially by promoting the oxylipid signaling pathway and leaving the plant in a priming effect that may be correlated with the increased resistance against Xylella. So I'd just like to thank all my lab mates, and a special thanks to my supervisor, who is also here, and thank you so much for your attention.
Thank you very much. The next speaker is Marie-Agnès Jacques from France, and she will talk about prophage diversity among French isolate of Xylella subspecies multiplex.
Hello, everyone. So I would thank the organizer for inviting me to present today, and it's my pleasure to present a work that has been done by our postdoc, Noé Arroyo-Vélez. And he has left the lab now, that's why I am presenting part of his work, and it was funded by the Big Xyl project. So Xylella fastidiosa has been introduced in France certainly or probably 50 years ago, and the subspecies multiplex is the only one being established in the country, mainly, well, I cannot show you, in Corsica, Paca area, and Occitania. And the ST6 and ST7 lineages are dominant over the two, ST88 and ST89, that have been detected only in few foci in Paca area. So while the control measures have limited the spread of Xylella fastidiosa in France, it still represents a threat to agriculture and the environment. That's why we wanted to have an idea on its evolutionary history and the capacity to evolve in the future. So we decided to focus on prophages. They are known to be major determinant of genetic evolution in plants through horizontal gene transfer. Indeed, during minutes after injection of viral DNA into a bacterial chromosome, the phage can opt either to initiate a lytic cycle or to transfer its DNA into the chromosome and to become quiescent. And in these later options, most of the genes, and including the lytic genes, are made quiescent do not expressed anymore, and the remaining genes are copied during the genome replication of the bacterium. And then many complex evolutive mechanisms are involved in the evolution of the prophage genes and in the chromosomal, in the bacterial genome. And some prophage may become domesticated, and then they represent only an evolutive tool for bacteria rather than being an harmful parasite. So we wanted to have an idea about the prophages that are present in fastidiosa subspecies and lineages to have better idea of the evolutionary history. So we sequenced by long-read technologies those genomes of 67 multiplex strains that have been isolated in France by our colleagues from ANSES. And this collection of strains harbor 24 ST6 strain, 41 ST7 strain, and one of each ST88 and ST89 strains. So the analysis of all these genomes revealed that all strains harbor prophages, ending in a collection of 517 prophages sequences that were analyzed for their taxonomy. A third of these prophages were complete, while the 2/3 were incomplete. And altogether, these prophages are in a number of 5 to 10 in the genome of each strain, which is quite a high number compared to what is known for other plant pathogens, such as Ralstonia solanacearum, for example, who hosts only 2.8 prophage region per genome. And the prophage represent an average of 9% of the Xylella fastidiosa genome, which is high, as the prophage represent from 150 to 400 kilobase pair in each Xylella genome. So they were analyzed for their taxonomy using various informatic tools, and this ends in 37 species clusters that are represented here in this tree. And these 37 clusters grouped into two major clades. One is the Codoviricets class of prophage, and the other one is the Lophivirus, a genus of virus. The Codoviricets were mostly incomplete prophages, so certainly have been introduced a long time ago, while Lophiviruses were mostly complete genomes. So certainly Codoviricets were ancient acquisition. And this taxonomy was confirmed by protein content. I will not develop today. And here you have the taxonomy of the viruses up in contrast to the taxonomy of the bacterial host. And in the column, you have the taxonomy of the Xylella strain. So in red, you have the ST7 strains, and in blue, the ST6 strains. And then in the second column, you have indication of their isolation area. So what you can see is that the pattern of distribution is really linked to the taxonomy of the hosts. So this is coherent with an evolution in allopatry, at least for this last amount of time. What is interesting is that Paca and Corsica ST7 strains have 2 Lophivirus and 5 Codovirus, as ST7 strains from the same area have 2 Lophivirus also, but not the same, and more Codivirus sets. Occitanie prophage, which appear at the base of the figure, are different to other ST6 strains, indicating separate introductions and, of course, evolutions in allopatry. and 2 codoviruses are conserved in most strains. And here, what I highlighted is a very closely related virus that certainly are orthologs. So this means that they were present in the last common ancestor of 2 strains and evolved separately after speciation, so differentiation of these sequence types. And you have also other cases where this gene certainly evolved in paralogs. So we wanted to know what was the global distribution of this prophage, and so we collected all genome sequences, complete genome sequences from NCBI database. And what you can see here is that the distribution, the pattern of distribution of prophage is very linked to the taxonomy of the strains, of the host strains. And if we focus on multiplex strains, what we can see is that codoviruses are very present in all strains, with 2 or 3 codoviruses present in most of the strains. So that certainly represents ancestral acquisition and codes for essential functions. LOFI virus are present only in European strains, in all French strains, and in a few Spanish strains. Of course, we wondered what was the biological role of the genes that were introduced by these prophages. So we made analysis with orthogroups. And of course, most of them codes for unknown functions. But for a few of them, they are very good indication. And here I will just highlight one which is interesting because the genes code for acetylxylan esterase, which is involved in the degradation of xylan, which is the main plant cell wall component. So it could be very interesting for the virulence of the bacterium through this phage acquisition. So I will end my talk with these take-home messages. And one very interesting thing, I think, is that this phage could have imported very interesting genes for the virulence of the bacterium. And I thank you for your attention.
All right, thank you. So the next speaker is Carlos Seyes Rivera from IVIA, Spain. He's going to talk about also phage diversity across Xylella fastidiosa, subspecies fastidiosa, and multiplex.
Okay, good evening. I think by now most of you already know what a bacteriophage is, but just in case, a bacteriophage is a virus that specifically infects Ankylosing bacteria. And when we talk about phage-based biocontrol, we are talking about exploiting these natural predators to selectively reduce harmful bacteria populations. Even though we can actually infect strains from different parts of the world, such as USA or France, our main work is, its target is the active mainland outbreaks in Spain, which are basically the outbreaking Cáceres caused by the subspecies fastidiosa ST1, which is infecting Vitis vinifera, Cistus, Lavandula, and other forestal species, and also the subspecies multiplex STC outbreak in Alicante, which mainly affects almond, but also it's been found in another 29 species. Most of you work with Xylella, so you know how hard it is to grow. That's why many studies that are trying to isolate Xylella phages rely on surrogate host strategies, which are basically based on using a phylogenetically close relative, such as bacteria from the Xanthomonas genus, for a faster screening process. It also has the advantage that you can work in lower biosafety requirements. And in that way, you also have a suitable production for host, so you can mass produce the phage for agriculture use. But it has the huge disadvantage that you have an uncertain success rate, meaning you really don't know how many phages are actually going to be able to infect and also control Xylella. On the other hand, we have the direct target approach. It's basically using your pathogen and trying to isolate directly in that. you have also a greater chance of finding subspecies-specific, ST-specific, and even trait-specific phages. And it also gives you a better understanding of phage diversity in general, which we will get on later. But it has, on the other hand, the disadvantage that it's much lower and more labor-intensive work. And you might encounter also the problem that your phages are so highly specific that it might be difficult to propagate at large scale. In our work, we actually used both the strategies. We were successful in both the strategies for the subspecies fastidiosa. We were able to recover the 25 phages using the surrogate host isolation technique. But for the subspecies multiplex ST6, we were unable to find any phage using this strategy, and we had a collection of more than 100 phages. So we had to use the direct target isolation. From the 17 I've noted here, only 7 were isolated in subspecies multiplex, the other 10 belong to the subspecies fastidiosa. Coming back to the diversity, we actually found high genomic diversity between the phages isolated in subspecies fastidiosa. They belong to 5 distinct viral families, but to note is the most important one would be the Autographibiridae family, especially the Pradovirus genus, which its name comes from the Pradovirus phage, which is Xylella phage isolated already in the USA in 2014, more or less, I think. So with previous knowledge and also our new sequences, we were able to propose in this paper 4 new genus and 9 new species. But what I really wanted to highlight is that 2 genera and 5 species were found only by phages isolated directly in Xylella fastidiosa. And for most of them, except the VC19, we couldn't find any Xanthomonas surrogated hosts. So we will have lost all this diversity if we didn't use the other approach. The same trend was also shown in the subspecies multiplex phages. For half of them, I couldn't find any surrogated host, which means I also couldn't produce them for agricultural use, which are the ones that I marked in black. And the main idea I want you to take home is that the optimal isolation strategy is going to depend on the final objective of your work. If your study is going to be based on ecology, phage diversity, phage typing on other uses, I really don't think the surrogated host is ideal. Because by selecting only phages capable of affecting a surrogated host, this approach is biasing the recovered population toward phages that have a broad host range, and you're losing all the phages only specific to Xylella. But if your approach is a biocontrol, then I think both are valid. But I want you to note that you will always need a surrogated host to be able to mass produce it in things like a bioreactor, because it's impossible to mass produce a phage only using Xylella. But of course, this is not the only thing you need in a phage to be able to use it for biocontrol. You will also need to make lots of in vitro assays to know the bacteria, phage dynamics, both in solid and liquid media. In our case, all the phages we tried, we saw for general dynamics. In some of them, the growth of the bacteria, which is the yellow line, was completely inhibited in the presence of phage. In other cases, we saw the appearance of resistances between the 5th to the 7th day. And in others, they were not that effective. We saw a doses dependency where high doses of phages were able to control the bacterial growth, but when you lowered the doses, they didn't work that well. And in other cases, the phage didn't completely inhibit the growth, just reduce the bacterial load. It's also important, I think, to know the host range of your phage using techniques such as the spot test assay with serial dilutions, so if you can know the efficiency of plating. Meaning I might have a phage that I have isolated in the ST6, it works really well, I have 10 to the 10 particles, but maybe if I try it like here in the ST51, the efficiency of plating, meaning how many particles actually infect and replicate, might be much lower. So I think this is also a really important test to do. So in a lab, we've already finished all the in vitro. We have selected the best phages that behave both in liquid, solid media, from different genus. And we have created cocktails for both the ST1 and the ST6, and are now trying greenhouse trials under control conditions. And also we have had the opportunity to do field trials in high incidence plots. For now, we are focusing in the ST6 for Prunus dulcis and the ST1, both in Prunus dulcis and Vitis vinifera, with both approaches, curative and preventive, and also checking if it also affects the rest of the microbiome of the plant. So I hope you find this talk interesting, and thank you for your attention.
Thank you very much. The next speaker, Ophir Bahar, will speak about the winter recovery of grapevine from Xylella infection.
It's the green one, right?
Yeah, green. It's also green.
Yeah.
We don't have the presentation on screen. Just wait. 1 minute. 1 minute.
1 minute. Okay.
So we're taking coffee.
Mm-hmm.
You do a dance or something.
Mm-hmm. No,
You sing. You sing.
If I want to have some questions, you want to ask me
If
something?
you have one question, Giuseppe, on the previous speaker. But your time is running. You were
No,
8
it's
minutes.
not.
No, I'm joking. What's your doing?
Sorry for the delay, but we don't know what else to do except wait. Okay, so in the meantime, remember that after we finish here, we're going to go to the poster session for a couple of hours. And we're going to have under the sun, so we're going to enjoy a little bit of the suntan from the area. And we run a little bit late, but again, we have at the end 30 minutes for questions, and then there's 2 hours for the poster session, so we can use the time here. Or okay.
It's okay.
Coming? All right.
Thanks.
There we go.
It's here, but it's not here.
But I don't see it here. All right. There we go. Okay. So, hello, thank you. And today I'll be talking about overwinter recovery from Pierce's disease in grapevines. And let's get started. So, Pierce's disease, as I imagine most of you or all of you know, is caused by Xylella fastidiosa subspecies fastidiosa, which colonizes the xylem vessels, as you can see very nicely here. And Xylella was first identified in Israel in 2017 in almond orchards, causing almond leaf scorch disease. And a few years later, in 2021, it was also discovered in vineyards, causing Pierce's disease of grapevine. In both cases, the sequence type is sequence type 1. And it seems like the primary vector that transmits Xylella in Israel is a spittlebug called Mesoptielus impictifrons, as you can see here in these pictures. From a recent report in California, you can see that the damage of Pierce's disease to the grapevine industry in California alone in 1 year could be over $100 million, which is quite a high penalty for a disease. And look at the situation in Israel. In these maps that you see here, every dot basically represents a grapevine. It's a little bit difficult to see because of the size, but each of the red dots represent an asymptomatic vine. So I don't know how well you can see it, but there's a lot of red dots in these plots. And the infection status in some of these plots can be as high as 25% of the plots. So it's a high infection rate in this area. And in an analysis that we did a couple of years ago about the effect of Pierce's disease on yield in these grapevines, you can see that even in the first year, designated here by symptom 1, you already have a significant decrease in the yield in both weight of the clusters and in the number of clusters. And then in the second year of infection, it goes even dramatically lower than that. So the effect on yield is very high, and it starts already in the first year that you see the symptoms. So combining the two together, the potential impact of Pierce's disease under Mediterranean conditions can be very, very high. One of the most interesting phenomena in Pierce's disease, at least in my view, is a phenomenon called overwinter recovery. This is a phenomenon that may be less familiar to people working with olives because it's primarily seen in deciduous plants, so plants that undergo winter dormancy. And this phenomenon is basically you have plants that are infected with the bacterium, they're showing symptoms, but for some reason at the following year they recover. They don't show any symptoms anymore, and the bacteria is no longer detected in the plant. So for some reason the plants manage to overcome the disease and become healthy again. These reports are coming on overwinter recovery coming primarily from California. And as I said, it happens primarily in deciduous plants, and it's associated again, mainly with low winter temperature, hence the name overwinter recovery or cold curing or cold therapy, that acronyms for overwinter recovery. So the question we wanted to ask, considering the situation in Israel, is whether the Mediterranean conditions, the Mediterranean climate can also support overwinter recovery to a certain extent. And the factors that we wanted to test is first temperatures and the time of inoculation, so when the grapevine is inoculated, and the two different cultivars with a slightly different susceptibility to the disease. So the way this experiment was set is in the following way. We selected two different genotypes, Chardonnay and Cabernet Sauvignon. From each plant, we had 50 grapevines in each group, and each group was inoculated at a different time point from May to September. And we kept them in the greenhouse and followed after symptom development and infection status. And then just as they entered dormancy, half of the infected plant were transported to the northern part of the country where the outbreak is, which is supposed to be a bit cooler than the central area of Israel. And they went under the dormancy in that location. And then when dormancy ended, the plants were brought back to central Israel, to the greenhouse. They were pruned and then followed for the persistence or recovery of symptoms in the following year. So this is just showing you an example of the symptoms that we could see in the greenhouse in the year the plants were inoculated. And this is the incidence of infection, just to show you whether the inoculation was effective or not. So you can see that in the earlier time points in May and in July, we had nearly 100% successful inoculation that also resulted in symptom expression. And then in the later ones, for example, in September, we did have around 60% infected plants, but with no symptoms at all. And a similar picture was also seen with the Chardonnay. Now looking at recovery, so what we see here is that the earlier the infection was, the lower percentage of plants that were able to recover. And on the other hand, the later the infection, if you inoculate them late in August or in September, you have a much higher rate or much higher chances of the plants to recover from the disease. And this was also, again, similar with Chardonnay. When we look on the correlation between symptom severity and the ability to overwinter, we see again a similar pattern. Plants that had no symptoms but were infected were the ones that had the highest chances of recovery, and those that had the most severe symptoms, as you see here, 5 and 4, had the lowest chances of recovery. But still, some of them did recover eventually. In terms of the two different locations that we tested, Central Israel and the northern part, we didn't see any difference between the two sites. I can tell you that the temperatures were not dramatically different between the two locations. It was only about, on average, 3 degrees centigrade different between the two locations. And so we didn't see any difference there. So the main parameter that influenced the overwinter recovery is the time of inoculation. Okay, so to conclude, we can see that the recovery from PD can occur under Mediterranean climate, under some conditions, and that early infections are much more likely to become a chronic infection, while later infections have better chances to recover. We've actually recently described overwinter recovery in a recent review that we wrote and just been published in Plant Disease. So for those of you who are interested, I encourage you to go and look it up. And we basically also propose a model for this where there's basically 3 different parts in the grapevine cycle that are critical to determine whether overwinter recovery can occur or not. The first part is spring and summer when the inoculation occurs. The inoculation can occur early, can occur late. It really depends on the biology of the vector, of course. And then the aggressiveness of the pathogen, the susceptibility of the plant, the climate will determine how vast the infection would be. The vaster it is, the less likely the plants will be to recover. And then the later, the second part is the fall and winter, where a lot of the inoculum is actually lost because leaves are dropped, all the pathogen in the leaves is gone. Winter temperature can have a direct effect on the pathogen. And of course, dormant season pruning will also remove large parts of the inoculation. So it really depends how much the bacteria were able to spread from the inoculation point and overwinter in high enough amounts in the main branches of the vine. And this will eventually determine whether the infection would become chronic or recovered. With that, I would like to thank all the people that were involved in this work, our collaborators from abroad, and of course the people who wrote the paper. And thank you very much for your attention.
All right, so now we move to the pitch talks. And the first one is from Felix Moran from IVIA, Spain. And he's going to talk about the tracing the origin of the new Xylella fastidiosa subspecies fastidiosa outbreak in Extremadura, Spain, through comparative genomic analysis and phase typing.
Thank you. So I'm going to talk about the last outbreak that we have in Spain, specifically in Extremadura, and the study of this outbreak. Oh, sorry. Okay, like everybody knows, Xylella fastidiosa subspecies fastidiosa is associated to the Pierce disease, and the detection of this lineage in Cáceres raises an important epidemiological question, like if this is a new independent outbreak, or maybe this outbreak is associated to another outbreaks that we have in Spain, or maybe in Europe. In order to answer this question, well, in this workflow, we followed in summary the PM7 protocol suggested by the European Plant Protection Organization, and all the isolates that we recovered from this outbreak were sequenced by hybrid approach, and also we analyzed using a phase typing approach in a last stage in order to know the virulence of this new strain isolate from the outbreaks, were inoculated in grapevine, and then evaluated by were isolated the isolate from the plant. Sorry. Okay, in this table you can see the summary of the genome of the strain genome of the strain that we isolate from this outbreak. In summary, we isolate 6 strains from different hosts, from Cystus, Cystisus, Ulex, and grapevine. One important thing is that we don't recover any plasmid assembled. We try to find some plasmid, but we don't recover any plasmid. The analysis of the core genome of these strains show that, in summary, that they group with the other strain isolate from the other outbreaks that we have in Portugal, which have sense because near to the Extremadura, and also with the strain isolates in the recent outbreak here in Italy. And curiously, they group together with strain isolate from US, specifically the strain Stagleaf. Okay, another curious result that we detect in this assembly is that we confirm the integration of a complete plasmid inside the chromosomal of this strain. We detect this integration and we confirm using PCR and Sanger sequencing. And also we evaluate in silico the strain isolate from the last outbreak in Europe. And also we detect this integration for the first time, which is curious because this plasmid is associated to the virulence of the strains. Also, when we evaluate the phase typing, we find a similar profile that we find in the core genome analysis. The strain isolates from Cáceres, specifically, they show a similar profile like the Stagleaf strain isolated from US in California. And another important result that the profile that we obtain with the isolate from Extremadura are very different to the isolate of the same subspecies detected in Mallorca. Regarding the prophase analysis, we detect some similar results about the profiles. The strain of Cáceres and Portugal and the Stagleaf show a same profile of the presence of prophase. The very different of this prophase detecting the strain isolate in Mallorca. And curiously, we detect the same sequence of the prophase in the Italy strain, but located in a different position of the chromosome, suggesting a probably a recombination event. And the last study, in order to know the virulence, we inoculate one strain isolate from the Ulex, Cystus host, and we inoculate in grapevine, and we detect symptoms after 92 days post-inoculation, and we recover the strain from this inoculation pathogenicity test with a successful result. And in conclusion, the result of this study showed a close relationship between the recent European outbreaks in America, the full chromosomal integration of this plasmid, which is associated with the virulence in Xylella, and the phase-based typing reveal a consistent lytic profile, which have in consonance with the results that we find in the core genome, and the systematic surveillance using the HTS of whole genome sequencing should be consolidated like a central tool for the detection and traceability of this pathogen. Thank you.
Thank you. So we continue the pitch talk. The next one is on a transcriptome of almond cultivar in response to Xylella with Giorgia Troncina.
So good afternoon, everyone. I'd like to share some preliminary results on early transcriptomic response of 3 almond cultivars to infection with different strains of Xylella fastidiosa. The aim of this work was to understand what happens during the early stage of the infections and to integrate transcriptomic data with SNP analysis to identify potential molecular markers associated with the early response. To do this, we inoculated Pizzuta and Tuono varieties with 3 different strains, as you can see. Unfortunately, Avijor was inoculated only with the strains ST1 and ST53 because some of the plant died. We did not have the necessary replicates to do the inoculation with the strain 26. We collected sample 48 hours post-inoculation, and moving into the results, let's see the principal component analysis. Here, the host genotype is the main factor driving the transcriptional variance. We can see in the PC1 axis 30% of total variance, and Pizzuta clearly separates from Avijor and Tuono, and it suggests that Pizzuta respond in a distinct manner at the transcriptional level from the beginning. A very similar trend can be observed in the mirror bar plot showing differentially expressed genes across multiple comparisons. Each cultivar displays a distinct pattern of down and up-regulated genes across different strains. In particular, for Pizzuta and Tuono, strains 26 triggers a massive transcriptional response, confirming that the host response is not only specific, but also also the strain is specific. So when we look closer at the geo-enrichment analysis, Avijor activates a response related to carbohydrate metabolic, cell wall degradation, or chitinase process. Tuono instead focuses on redox regulation, on transmembrane transport, so with less evidence to specific defense pathways. Pizzuta is enriched in functions related to cell wall degradation or calcium ion binding. And through this RNA-seq and SNP genotyping data, we identify several high confidence genetic variants and within differentially expressed genes, and some of these SNP show a specific strain pattern. So our preliminary results suggest distinct strategies adopted by the cultivars following the Xylella fastidiosa infection. In particular, Pizzuta and Avijor exhibit a more coordinated defense involving, for example, in pathogen perception, defense signaling, peer protein activation, or cell wall remodeling. But Tuono shows a broader stress-associated response, so shows a less evident coordinated defense activation. Our preliminary results, so based on our preliminary results, we have to do hard work, but next step will be to extend additional time point to validate candidate genes and identify robust molecular markers. So I would like to thank EFSA for supporting my travel and my stay here, all of the co-authors, the Institute, and the project OmiBread. Thank you for your attention.
All right, our next speaker is Leticia Lucero from CNB, CSIC, Spain. And she's going to talk about exploring the potential of Rep WH1 prion-like proteins in the control of the phytopathogen Xylella fastidiosa.
Okay, thank you very much for the introduction. First of all, thanks to the funding to the EFSA for funding my stay here with the Young Researchers Initiative. And I want to start by presenting the main actor of my work and of my lab, which is protein rep A. This is our replication protein from a plasmid in Pseudomonas sabastanoi, and we saw that upon the isolation of one of its domains, the wing helix 1 domain, this one, when it's expressed in a model bacteria, Escherichia coli, is able to form aggregates that are toxic for this bacteria, forming amyloid aggregation and finally the cell death. So we were wondering if this replication protein is also present in, well, it was present in other plasmids present in Xylella fastidiosa. We found more than 70 homolog proteins in different subspecies from Xylella with more than 40% of identity. We decided to select 2 of them that were in 2 representative groups. One of them, as you can see it in the screen, is called wing helix 1 PXF51 in yellow. You can see it, and it belongs to subspecies Pauca. And the other one is wing helix 1 PXF64, which belongs to a plasmid presence in species multiplex. So we will be working with the 3 proteins, the 2 from Xylella and the 1 from Pseudomonas sabastanoi. So we expressed each of the proteins within Xylella fastidiosa, subspecies fastidiosa, Evia 5235. And, well, a disclaimer, I might say that the multiplex protein was unable to be expressed within this Xylella fastidiosa, most probably due to its high toxicity. If you want to know more about this, I'm not going to go further into this protein, but you can come to my poster, which, by the way, is number 53. So the protein belonging to subspecies, Pseudomonas sabastanoi, as you can see it in blue, was able to form aggregates within Xylella fastidiosa. And in the case of the protein that belonged to subspecies Pauca of Xylella, you can see that it was apparently soluble within the bacteria. And I say apparently because when we did a dot plot assay, again, if you want to know more about this technique, you can come to my poster. We saw that in the 2 cases, the protein was able to form aggregates that, furthermore, in the case of the protein belonging to Pseudomonas sabastanoi was of amyloid nature, as we demonstrated before in Escherichia coli. So what happened when we injected the bacteria expressed in the different proteins in planta? So we saw that there was a diminishment of the symptoms in the plants upon 3 to 4 months post-inoculation. What we saw was that in the first place, when we induced the expression of the proteins in vitro, as you can see in these transmission electron microscope images, the cells the cell membranes were completely disassembled. When we go to the images on transmission electron microscope, in which you can see it in the right, we did a section at month 3 post-inoculation of the plants, which, by the way, are Nicotiana bentamiana. And what we saw was that the symptoms were still present in the bacteria, and these were shrinked and in a bad health. So we think that because there's not, after 3 months post-inoculation, there's no presence of the antibiotic which makes the plasmid that expresses these proteins to be present, we think that this is because the presence of the symptoms in the bacteria are still because of a hypothesis that is the prion hypothesis, which says that you don't need the plasmid but only the protein that promotes the assembling into aggregates of the self-replication protein present in the plasmid of the bacteria that we are using. Well, thank you very much for your attention, and if you want to know more about this, you can come to poster number 53.
Thank you. The next speaker is from ANSES, Victor Nelaton, or Nelaton, I don't know how to pronounce, sorry. He will talk about the genetic diversity of Xylella in Occitania.
Okay. Hello, everyone. So, yeah. I would like to thank CHM Bari for funding my participation to the conference today. And I will talk about the genetic diversity of Xylella fastidiosa in the Occitania French region. So first of all, why are we studying Occitania? Because it is the most recent French region to be shown infected. And we have so far no knowledge of the origin of the contamination. And as you can see on the map, so every spot is a sample, and the red spots are the positive samples during the official surveillance. The area covered by the positive samples is quite large, and this is a kind of new situation for France because in Corsica it is an island, so it's delimited by the sea, and in Paca it's mostly along the coast, and here it's within the land and it's quite large, so we would like to have more information. So what am I trying to do in the first part of my PhD that I'm presenting today? First, we want to confirm the presence of only ST6 and ST7 in this region. Then we want to look if the strains in Occitania are related to other strains either in France or Spain. And as ST6 are also present in other regions of France and Spain, and ST7 in other regions of France. And also, the final goal will be to highlight the fine intra-regional genetic structuration of the Occitania strains. To do that, we are using a method called MLVA, which is a genotyping method based on what we call VNTR. There are markers that are microsatellites or mini-satellites. And the scheme we are using was developed by a former PhD student in INRAE in France and ANSES. So we typed 331 samples from Occitania. Then, with the results, we are using clustering techniques to try to find some clusters in the strains. And then we are trying to use other data, for example, the host plant or the geography, to find some characteristics of the clusters. But for now, it's only a preliminary visualization. So the few results we have for now, within the 331 allelic profiles, we got 326 ST6 strains, so a vast majority that are all across the region, and only 5 ST7 strains that are really locally distributed. It's a restricted area. What we also found, and to see the first two points of this slide, I invite you to go see the poster. The poster is the number 57. So the ST7 population in Occitania is genetically similar to other French ST7, so it indicates a probable inter-regional transfer. We don't know in which direction, but probably. And then for the ST6 strains, it is a new cluster. So it is distinct from French ST6 and Spanish ST6. So it's probably an independent introduction, and I think it's quite interesting that it shows the same thing that Marie-Agnès just explained before. And the only thing that I have here on the slide, for the other things you can look at the presentation, is that within the ST6 strains of Occitania, we can find 2 subclusters that are like clonal complex. And the question that still remains for now is, are they distinct introductions, or are they the product of on-site diversification? If you want to see how these strains are on the map of Occitania, you can see the poster. It's on the figures on the right. I thank you for your listening. Thank you.
So the last presenter of the session is Elif Olkun from the Ludwig Maximilian University of Munich. And she's going to talk about HFQ-dependent small RNAs mediating environmental sensing in Xylella fastidiosa support lifestyle transitions and virulence.
Thank you very much. As you might all know, one of the hallmarks of Xylella fastidiosa virulence is to switch between the planktonic growth and the biofilm in response to environmental cues mediated by quorum sensing. And during this time, it is known that Xylella releases significantly higher extracellular vesicles, which is already known to be a hypervesiculating bacteria compared to its closely related species. And when we categorize the cargo of these extracellular vesicles, we found some non-coding RNAs and also a protein called HFQ in the extracellular vesicles. As you can see in the electron microscopy image, we immunolabeled HFQ and found that it is highly associated with the extracellular vesicles of Xylella. So what is HFQ? It is an RNA chaperone protein and is a central regulator of RNA metabolism in many bacteria, which regulates the biofilm formation, the virulence, the twitching motility in many bacteria, and it facilitates the base pairing between the small RNAs, the non-coding RNAs, and their associated messenger RNAs to mediate the post-transcriptional regulation in bacteria. So what we want to understand is what is the role of HFQ and its associated small RNAs, and also in Xylella virulence, and is also in association with the extracellular vesicles in the means of communication between in the microbe community. So as a first step, what we did was knocking out HFQ to see phenotypic changes, and immediately we were able to see that HFQ mutant is less motile, produces more biofilm markers, and also it's deficient in producing planktonic growth, and also reduced in cell motility. And when we also infected some model organisms like Arabidopsis thaliana and tobacco, and here grapevines, we were able to see that HFQ mutant compared to the wild type is reduced in systemic infection of the grapevines. So it was not able to travel along the plant as much as wild type. So when we wanted to understand the more molecular mechanism of HFQ, so how it's regulating this, this phenotype and the phenotype that we see in the plants, what we did was conditioning the wild type and HFQ mutant to xylem mimicking medium, and then doing transcriptomics to see differentially expressed genes as a sudden response to this xylem mimicking condition. And we were able to see that in HFQ mutant, type 4 pilus genes or transcriptional regulators and signaling responses associated with quorum sensing and biofilm were impaired. And also, since HFQ is regulating these genes via small RNAs, we did small RNA sequencing and did a de novo small RNA annotation of Xylella fastidiosa in both conditions of rich medium and xylem mimicking medium. And now we have a novel HFQ-dependent small RNA candidates that might be mediating the gene expression relating to biofilm and quorum sensing, twitching motility, and virulence. So with that, I would like to thank my lab and the organizing committee and everyone listening here. And if you want to know more about HFQ, you can find me in poster number 73. Thank you very much.
So now we have time for questions. Yes, Pasquale.
Thank you. I have a question for Carlos. You mentioned a preventive approach of using bacteriophage. What do you mean? How do you intend this preventive approach?
Yeah, okay, now. What I mean by preventing is basically introducing the phage before the bacteria in the plant. That's easier done under greenhouse conditions. In my case, these times I'm giving it 2 to 3 weeks. I've tried endotherapy with pinpricking, spray, and also irrigation. So I'm trying the 3 things at the same time because I have a limited time. I'm still doing my PhD, so I need the results within a couple of years. And basically, after 2 to 3 weeks, I'm going to inoculate the bacteria and check if the phage is still able to control it.
Yeah, but when you inject and there is no cells that host and replicate the bacteriophage, how did it survive?
I think I know what you mean. So the phages are stable, like broad range of temperatures. I don't know if they'll be stable inside the plant, which is one thing I'll have to control. That's why I'm trying to read all approaches, like curative, preventive, different measures at the same time. For now, I don't have those results because I've just started the implant assays, but hopefully I'll be able to respond to you in a few months. But for now, they are stable at different pHs and temperatures. Normally, it's protein capsid. It's not that sensible to temperature. The only thing that makes it more sensible normally is UV rays. But since I'm trying to get inside the xylem, I don't think that's going to be a huge problem for now.
Question? There's a question here.
Thank you. This is a question for Ophir about the winter recovery, because you mentioned that probably it is explained by cold effect during winter and also dormancy of the plant. Are there more possible factors affecting this decrease or even disappearing of the Xylella from the plant?
Thank you for the question. So the short answer is yes. I think there's many other factors that probably determine whether a plant can overcome the disease or not. I was mentioning that it was primarily associated with cold winter temperature because these were the first observations and the first experiments done on overwinter recovery was to expose the plants to different temperature for different durations, and they could see that there is an effect for the temperature. But looking at all the current data on this and other pathosystems that also undergo overwinter recovery, I think it's maybe one of the main factors is how established the pathogen will be when the plant reaches the dormancy stage. If it's not well established, then there's a high chances that some of it will be just removed, either by leaf dropping or by the pruning. And then there's also an option that there's a part of it that's related on the plant response after the dormancy stage, which is still at the hypothesis level, but there are indications that it's not only about cold temperatures that are harming the bacteria, but also maybe some kind of a more effective plant immune response after dormancy.
Questions here in the front. Alison.
My question is for Lena. I cannot see her. But okay. Lena, I saw in your talk that you showed the symptoms in Nicotiana tabacum. That for me was very interesting because you showed that when we have plants standing, it means that the plant is, the symptom is more severe. And I also found it in my lab, but I couldn't, related these symptoms with severity. And probably it is, probably you are right, and I didn't see that in my experiments, because I have different symptoms in the Nicotiana tabacum, like you showed there, but also was very curious for me when you related the plant study with more severity. So that's nice. But my next question is, do you also correlate these symptoms with the bacterial colonization or population, with the symptoms, or only visual symptoms?
Thank you for your question. I did not do a correlation between symptom severity and the bacterial load. Actually, all the bacterial load were pretty low with pretty high CQ. So we only compared the CQ with the control plant and the symptoms with the control plants, but I did not do a correlation between symptoms and CQ because they were all really high.
Other question? Yeah, Pasquale, please.
Question for Alessa. So can you imagine a post-translation gene silencing strategy to protect from Xylella as it is from plant viruses?
Yes, currently I'm doing very basic research, but in the long run, I think it can also lead to some RNA silencing methods, especially, for example, mediated by delivery through extracellular vesicles or with the help of HFQ protein. So when they deliver to the bacteria, they can silence some of these genes, either the small RNAs that are regulating or the messenger RNA directly, so that they can actually reduce the systemic infection or growth of the Xylella. But currently, yes, it's in a very basic research state. But that's what we also want to be able to, that's why we are researching this system also, yeah.
Any other questions? Okay, I have a question for Marie-Agnès Jacques. So when you look at the prophages, you see like a relationship with the origin of the strain, yeah? It seems to be like separated. So why do you think is that? Do you have any hypothesis of why the prophages are related to the area?
Well, we tried to see if there was a correlation. Apparently it does, and I think it is coherent with an evolution in allopatry. And for most of, at least introduction in Europe, there were separate introductions and populations evolved separately, the one for the others. So finally, they may have acquired prophages from, phages from their environment, which are specific. And so that's what is clear, especially for the lophovirus that were found only in European strain, most of French strains and a few of the Spanish strains. I have to look closer to see if in Spain there are really closely related strains in the term of space. I haven't done that so far, but I think it could be very interesting to confirm this hypothesis.
Any other questions for the speaker? So one general question, since we have time now. So anybody can answer this. So why do you think Xylella is so difficult to control? What is the key thing that we need to understand that we don't know?
What a question.
The one with the right answer gets $1 million. Paid by Giuseppe from the EFSA funding.
Well, I think for me, my main worry or problem is actually reaching Xylella with the phages, for example, because it's a pathogen that we know is the xylem, so it's, it can go to the roots. So for me, my main worry, I think one of the most difficult things regarding Xylella is actually reaching where Xylella is with your preventive measures, being peptides, phages, or any other compounds. I think that's one of the most challenging things we're having in all our measures. Control.
Any other ideas or comments? No. One second.
Your question is very difficult. I think what would be interesting is also to have your answer to your questions. But
That's not fair. I ask a question.
No, but for me, yes, I think I agree with you. It's very linked to the non-homogeneous distribution of Xylella within the plant. So especially when you face an early infection in plant, it's very difficult to really precisely see where the bacterium is and so to control it. And also at the level of the bacterium, so at the scale of the bacterium, it's really difficult to identify the vessels that are infected. So it makes me more difficult, the control of the bacterium, of course. And then you have all the different physiological stages that the bacterium is also experiencing.
I mean, also, what came to my mind is that it's a very small genome size. It's as if that it's very specialized to grow in a very specific environment and only in that environment. So it might be also perfectly growing there. So it's very hard to get rid of this in that very specific environment. So with the reduced genome size, it seems like it's very, targeted itself to grow in a specific way, but very strongly. So from my experience, it feels like that it also might be correlated to why we are actually cannot really target and get rid of the bacteria from the xylem.
Anybody else from this side?
Too much light, maybe?
Valeria, good?
No, it works. Okay, according to the great plasticity of Xylella genome and the different capacity to adapt to the different hosts, I asking you how you manage in America this pathogen. Which is your approach? Because
to manage
to control, I don't know, is how you manage the Xylella disease.
In the US, the only thing people are doing is insecticides, mainly in California. It's just insecticide and removal. In blueberries, they just remove the bushes when they are infected, and that's it. They're not using insecticides in blueberry right now, but mainly in grapes in California, yes, they use insecticide. But then, yeah, it's endemic, so we don't care, or we don't care. We're not looking at where it's expanding because it's everywhere pretty much already. So it's not like you guys are there, you're trying to contain it and not move to other places. In the US, it's too late. It's everywhere in the US that the weather, the climate allows. So yeah, the only thing is insecticide. There's some commercial products for phages and stuff like that, but for what I heard, it's not been successful. They don't work well. So the only control you can do is insecticides. Any other questions or comments? For the waking up, that's good. Wait a second, there's somebody here and then Emilio,
Okay.
1 second.
I have a question for you, Leonardo.
Okay.
Thank you for the work on, with the correlation between the biofilm and the symptoms, but in some cases there is no correlation. How do you explain when you have biofilms without symptoms?
Okay, so you're saying the lack of correlation between the symptoms
Yes.
and yes. At this moment, we think it may be, you know, the biofilm part is strain specific, that most of the strain that we tested, biofilm is important, but maybe not all of them. Maybe some of them, they cause symptoms, you know, not by blocking the silent, but by producing some signal compounds that do that. But yeah, we don't know. Like, interesting, the Temecula, the model system wasn't following the biofilm, more symptoms. You know, there's been this debate of like, oh, is biofilm important or not for disease? And that debate has been based on work on Temecula. So Temecula, in our hands, it doesn't follow that rule of more biofilm or symptoms. So maybe in that case, it's something different. But in most of the strains that we tested, it is more biofilm, in vitro, with SAP, correspond to symptoms. So it may be, yeah, at this moment we're thinking it's just like different mechanism of virulence that we don't know. There was something from
Emilio?
Yes, I think this is for Maria Agnes about one of the comments you tell about, you said it is expected that the first introduction in Occitania or in Corsica was 50 years ago. What is the basis of that and the fact that the first detection in Europe was in 2013? so there is, it's the same in Spain, in Mallorca, because people say maybe more than 20 or maybe 30 years. So what has happened between this time and the first detections? It's difficult to understand that.
Thank you for your question. We made earlier on different analysis on the genomes, and we date the date of divergence between the French ST6 and ST7 strains from their American counterparts. And the date we identified was the year 1973 for ST7, I think, and a few years later for 96. So it makes nearly 50 years ago from now. And so this is based on the divergence between the genomes. And the same date was found by a colleague, Samuel Soubeyran, who used some mathematical modeling based on the repartition of plants that were sampled positive and negative in Corsica, taking into account the existence of an habitat that was not sampled, and he found the same probable date of introduction of Xylella in Corsica, so around 1970. Okay, so that's the date. So what's happened during 1917 and the first detection in 2015 in Corsica? Well, just that the symptoms were teeny symptoms. At the beginning, they were mostly seen on ornamental plants, mostly Polygala myrtifolia, and then in many plants in the Corsican bush. But then, you know, if you have only a few plants that show a very few symptoms, it's very difficult to detect them and to sample. And they were just not identified as linked to an infection, but more linked to stress, to drought. So that's certainly the reason why it was never sampled. And if someone were, made the detection
possible for the first polygala in Corsica, it was because it was somebody who was aware of the detection of Xylella in Apulia. So he was really looking at plants that could show and present this type of symptoms.
Right. Any other questions?
We have one slide to show you from on Xanthomonas before we close this session.
Yes, if we can show the slide with advertisement. So next year in the summer, I think in June.
We don't have the slide yet.
Hopefully the slide will come while I'm talking. They're going to have the Xanthomona Daisy meeting, the genomics. So people interested in learning more and presenting. So Valeria Scala is here, and she's the organizer of the meeting in Rome, June 2027. So again, if you have any questions about the meeting, there will be more information in Rome, yeah, in late June. And the slide will come sometime, hopefully. Okay, good. Thank you. So now
do you want to say something about it? You can come if you want. We have time. We have 3 minutes and 50 seconds.
Good afternoon. It's a pleasure for me to tell about this Congress that we bring again in Europe, because the first time was in Germany in 2018, and then move in US in Florida to Nianban, and then in Shanghai, and now come back in Europe. So it's a very pleasure for us to guest all the people interested in this conference, and we'll be in Roma. That is a very nice venue.
Thank you. So we'd like also to thank all the speakers and all the questions. Also, thank you. We close this session. We have a poster session during 2 hours before the networking reception.
Okay, so go and enjoy the posters, talk to the students, and learn more about Xylella.
Yeah. Thank you.